Hello everyone present here and everyone who's following us online. Welcome to Premurosa mini conference, Precision Medicine, how far we are. And we're starting with Andrea Kokis, who will tell us what happens when an implant is infected. Please.
Okay. Thank you very much. I hope that everything is working. I feel like being in the moon, something like this, but it's okay. So thank you for the introduction. So today I will try to convince you that we will need to work a lot to prevent infection to the implants, which is a part of course, Premurosa project. So first of all, I have to convince you that infection is a huge problem from a clinical point of view, because I'm pretty sure that if I tell you cancer, if I tell you cardiovascular diseases or neural diseases, of course, in your mind, you always have a sort of red light, no? Danger. But if I tell you infection, maybe most of you say, well, okay, infection, when I'm sick, I take antibiotics, 5 days, 7 days, then I'm okay. But unfortunately, the situation is changing a lot. And in this slide, you can see the provision from NIH, so from the American point of view, which tell us that due to the ability of the bacteria to adapt to the drugs, within 2050, it's not so far, 25 years, okay? As you can see here, infection will cause more death than cancer, because we are working and improving a lot in some diseases like cancer, like cardiovascular diseases, but we forgot about infection. So soon, infection will be the first cause of death in the world. But why it happens? As I told you, maybe most of you is thinking, but why? We have antibiotics, it's easy. We can easily face infection. No, unfortunately, it's not like that. And it's our fault, because we are still using old drugs, active principle that comes from the 40s, from the 20s, from the 60s. And so in these decades, bacteria learn how to escape from the effect of antibiotics. So if you look at this cartoon, actually imagine to deliver your drug, and what happened? In order to be effective, we need that the drug interact with the bacteria. No? They attach to each other. And then what happened? Normally, the drugs enter into the bacteria and kill it by reducing metabolism, by denaturating DNA, and so on. So what happened in these years? It's since bacteria know our active principle, because as I told you, they are old, they understood how to block this binding with the drug. So the drug do not interact anymore with the bacteria. Or if they do it, so even if the drugs enter, you see bacteria change their metabolism. So the drug is not anymore effective because they have a new metabolism. Or they develop some pumps, so some mechanism that catch the drugs and kick it out. So it's impossible to have the effect. Or in some cases, we have even a genetic evolution, meaning that from a DNA point of view, bacteria are not anymore the same, so the drugs are not anymore effective. So this is the first problem. Bacteria, especially those ones in the hospital, are extremely resistant to the drugs that are now available. The second problem is related to this conformation of the bacteria, which is normally known as biofilm. Because what happens when bacteria attach to the surface of an implant or of a tissue, whatever? It's not just a single colony, because you have to imagine that this blue line is our implant, our tissue. So the first colonizer bacteria that are here, you see, they like the surface and they find proteins that act like a glue. So they attach because they have these proteins that link them to the surface. And then what happened? They start to stratify. You see in the cartoon how many layers they produce, and day by day, week by week, they grow and they form this very huge three-dimensional structure where they are really protected inside a sort of envelope. And again, even if you have a very effective drug, okay, what happens is that if you administrate a drug in such a structure, you can have an effect on the upper layer. If you're lucky, you can have an effect on the core, but you will never be able to eradicate this first colonizer that day by day continue to build this sort of house of bacteria. So every time that you apply the drug, but they continue to grow. And when they grow, they, you see, release some signaling to other bacteria. It's like, come here and colonize more. So when we have the formation of the biofilm, unfortunately, we cannot anymore eradicate with drugs. We have to mechanically remove it. When you go to the dentist and you do polishing, it's a little bit painful because it's a mechanical removal. Okay? So this is an example. So from a clinical point of view, just to give you an example in dentistry, what I told you, unfortunately, when we have an implant, the story is exactly the same. So here you see a healthy situation when we have the classical implant in dentistry. So one part is in the bone, then we have a transmucosal part, which is in the gum, and then we have the crown, which is the aesthetic part. When we have bacterial infection, normally we start from here, from the gum, maybe when the sealing is not perfect, then the bacteria, exactly as we saw before, start to attach, start to grow. And so at the first stage, we have a high inflammation, which is called mucositis. And here you see the gum that is highly inflamed. But unfortunately, as I told you, since we are really unable to treat the disease, if it's progress, so you see again our cartoon that the biofilm is increasing, started to release signals, what happened? That we move from the gum to the bone. And this is exactly what you see, because you see that the bacteria try to start to destroy the bone structure, and now you see even the screw, because it's really, the tissue is degrading, it's destroying. So this is the huge effect of the biofilm when it's grow. It produces toxins, it produces enzymes, everything which is really, really harmful for the tissue. Again, maybe you can ask, but why it happens? Come on, surgeons work properly, and what we expect is that the tissue attach to the implant, not bacteria. Come on. And this is hopefully true, because infection, of course, is a side effect, it's not a normal procedure. But unfortunately, if some bacteria enter, even if the tissue is doing the self-healing by itself, there is a sort of competition between bacteria and cells. If they are together in the same environment, they really fight to infect the same surface. So there is really a competition, bacteria versus cells. But normally, unfortunately, are bacteria the winner. Why? Because they're faster. They are better. They can adapt faster and in a better way to the new surfaces than cells. The cells require more time. Then we have inflammation, so it's not immediately a healing procedure. So unfortunately, if we have entering of bacteria in a surgical site, normally we have a huge infection. How we can face this problem? Of course, we have many solutions. We have no time to discuss all of them. But of course, we can work on the initial phase of the adhesion, so we can try to prevent the adhesion, so we can modify the surface of the implant in order to prevent the starting adhesion. We can try to disaggregate the biofilm when it is an early biofilm, meaning that we have few layers, so we have still time to remove them by drugs. Or if we have a mature biofilm, unfortunately, we have to think about something related to really dispersion or mechanically debridement of the biofilm. Because actually, as I told you, when we have a mature biofilm, then we cannot act again with drugs. Of course, in literature, you can find many solutions, but here I want just to show you something that comes from the Promurosa project, of course, in terms of infection treatment. So this is a nice review from Ljubljana that discuss about the possibility to use a particular metal, which is gallium, in the surface of implant. This is very interesting because gallium competes with iron. But bacteria need iron to survive because they need iron for the metabolism. But since gallium and iron enter in the same site, if gallium enter in place of iron, then the bacteria die because they are unable to use gallium as for their metabolism. So this is interesting as a sort of Trojan horse activity, like in the computer when we have a secret sequence, and then we act on the metabolic activity. Second idea that's come from the work from Mari, and this was the idea to put metal ions on the surface of bioactive glasses. Bioactive glasses will be largely discussed by Enrica Vernet, so I don't use this. Metal ions are interesting because they attach to the membrane of bacteria. They cause pores, so they destroy the membrane. And after that, they enter inside the bacteria structure. They destroy also the DNA. So it's a strong activity, and it's hard for bacteria to react towards these ions because it's not a specific binding. It's just the membrane binding. Okay? And it was published, and you see the results in comparison between not treated and treated. We have really few bacteria. Then another solution, and comes again from Maria and from Virginia, that are here, is the use of peptides, because antimicrobial peptides are structures that are normally produced both by cells and bacteria, and they are developed and they are released exactly to fight infections. Maybe for you it's not very familiar, but if some of you work in the food environment, food industry, I don't know, this name, nisin, should be interesting because nisin is used to preserve food from bacterial and yeast infection. So the nice idea in this publication was to use this particular peptide from the food industry to the implant, because if we put nisin on the surface, what you see is that we let's say, hinder bacteria to form this biofilm structure. So they are here, but they are in single colonies. Then we try to evolve this idea again with Mari and Virginia. And so we use the same antibacterial peptides, so again nisin, but we combine it with nanotopography. Why? Because if you look at the cartoon, you see that those particular topography of the surface, it's like a needle. I think that none of you love needle when you do blood collection because it's painful. So the same for bacteria, because when bacteria try to attach on the surface like this, they are killed because the needle destroy exactly as like your skin. It's not nice. They destroy the membrane. So in this case, the work was much more successful because we combine the activity of the peptides, which is a biochemical activity, with this mechanical disruption of the bacteria. And as you can see, we have a very nice reduction of the bacterial infection onto this titanium implants. And this was very successful. So, of course, these are some examples that comes from our project. There are many other in the literature. But since we have a wide public today, what I hope is that in the first part of the lecture, I convince you that unfortunately infections are a huge problem and we have to really quickly and strongly face them in order to prevent this disaster from a clinical point of view, which is very close. Unfortunately, implants are not an exception because bacteria like the material that we use for implants, metals, ceramics, polymers, whatever, they easily infect them. So we have the same problem. So also for implants, which is one of the focus of Premurosa, we need to improve from this point of view. And finally, thanks to the work of the Premurosa student, we have some new solution that merge, you see, some different idea. So last slide, just to thank all of the guys. It was really a pleasure to work with all of you. We enjoy, we also will be very productive. You see that when people work in biology, they are sad because the cells are complicated, bacteria are complicated. So Ilyana is still crying from the work that we did some months ago. But we did a great job, and I'm sure they all have a great future. So thank you for the attention. Okay, if you have some curiosities. Yeah. Ah, okay. Okay.
Hello. Okay, if you just go a couple of slides back with this, this one.
Yep.
Next one.
Next, this
Yeah.
one. Sorry. I have
It's
to
okay.
Yeah.
With nanopillars, you said that they rapture the bacteria, but how is it with connection with our cells then?
With, sorry?
With our cells?
No,
They
it's
grow
different.
over-
Yeah, because
Do they also kill our cells, so they cannot really
no.
grow over it?
It's the point is that imagine the structure of the bacteria. Of course, if you have a flat surface, no problem, because it's just like a drop of water. But when they face a structure like this, they have to stretch the cytoskeleton to adapt to the surface. Sometimes, not always, but sometimes it ends with the rupture of the membrane because it's not easy to adapt it, especially if you have gram-negative because you know the membrane is more thick. But one of the key of this strategy is that this nanostructure are sensed by bacteria because they are very small, 1 micrometer, 2 micrometer, but not from cells because they are bigger, 10, 20 micrometers. You know the strategy of Fakir, when they have the nails, and so it's not sensed. So basically, it's just sensed by bacteria, but not by cells. That's the trick. Yep.
Yeah. Thanks. Question. You are using, say, not.
I think we are online, so we have to use these complicated tools.
So you are using not a personalized
No.
approach.
Yeah, that's true.
What would be presently a better or a more approach with better efficacy?
Yes.
Because here it's it's random and a lot of bacteria not respond. Or,
yes, that's true. That's true. This is $1 million question, how to improve this, because of course I show very general. Phage maybe, this is one solution, but I think that at least from our experience, the most advanced technology that we are used to understand what is going on in this very resistant strain is to use sequencing. We want to understand from a genetic point of view why they are resistant from this point, from these drugs. So from this work, we are understanding that unfortunately some of these strains are a sort of soup of different strains, meaning that there is a part of Staphylococcus, Escherichia coli, Actinidobacter, and so on. So one of the let's say, to sequencing, we are able to design specific drugs that specifically bind from genetic point of view to inactivate, for instance, their activity. So at least from our experience, this is something that is pretty interesting. The other things, yes, is to maybe use some solution that comes from nature, like phages, but also antimicrobial peptides are interesting because also cells produce them. So yeah, it can be, from one side, we need to know better about the bacteria. From the other side, maybe, yes, we need to use something which is more effective, let's say, targeted manner. Yeah, sorry.
Thank you very much for a nice presentation. So I have one additional question regarding the material, gallium, so that was suggested.
Sure.
What about maybe potential concern on toxicity, the long-term release?
Yes, yes. This is a question that it's true for all the metal ions, not only for gallium. Unfortunately, we have to respect a sort of threshold where we have antibacterial effect but not toxic effect. From gallium, we are a little bit lucky because the threshold is higher than, for instance, copper, which is much more toxic and is used a lot in the literature. And gallium is very interesting, and consider that I think in the United States there are the first clinical trials from patients with third-degree burns, and also I think with other kind of diseases it's very effective. So the toxicity, of course, should be considered, but for gallium it's not so high like copper or silver. I don't say silver, but like metal ions. But this is a problem for all the metal ions. Also the biodistribution. When you use ions, unfortunately, if you look in the organs, you find some traces. Yeah, but it's a very good point. Yes, of course.
Any more questions?
Just a quick comment.
If I don't destroy it.
Thank you. I think Andrea was mentioned the gallium, and I think that's we still need to understand the fantastic results which come out of there, because it's very close to iron, as Andrea explained, but it cannot be reduced as an iron. So we can fake the bacteria that they think they eat iron and they try to reduce it, but it doesn't work. And because of if the toxicity will be managed, I think that will be very good non-antibiotic way of combating the bacteria metabolism, which can be, very, very revolutionary, which I can say in terms of how do you treat these kind of infections.
Thank you.
Thank you.
Okay.
Thank you, Andrea, for
Welcome.
enlightening the importance of
Yes.
safety of implants and the possible solutions. So now I will welcome the next speaker, Jasmina Stojkovska from Faculty of Technology and Metallurgy in University of Belgrade, where she will introduce how to do reliable drug testing on 3D models for beginners.
But this is your I have to do that. Okay, okay, I didn't know that. Okay. Yeah, this is my, I think that they will. Okay. Thank you. Okay, good morning everyone. I'm Jasmina Stojkovska, Senior Research Associate at Faculty of Technology and Metallurgy, University of Belgrade, and I am working in the biomedical engineering laboratory. I'm super excited to be here today. Thank you, Leah and Sasha, for this opportunity. And today, let's say I will be your tour guide as we explore journey of new drug discovery from lab right to the patient. So imagine that you wake up one morning, that you don't feel well, that you have a fever, that you have a very bad pain in your throat, and you go to the doctor. Doctor checks you out, does some tests, and conclude at the end that you have bacterial pharyngitis. And because of that, the doctor will give you drugs, antibiotics for infection, of course, and something for a fever. And after a few weeks or after a few days or weeks, when you finish therapy, you feel well, you don't have a fever, bacterial infection is gone, and you are a healthy person again. Unfortunately, some of diseases are incurable because we don't have drugs, or we have, but they are not so efficient, such as the diabetes, multiple sclerosis, or cancer. Therefore, the millions of people are still waiting for a new therapy. So the question is, why do we don't have drugs for this disease? On one hand, we don't know what causes these diseases. For example, what caused the cancer? Is it the stress? Is it the toxins? Is it the trauma? Or all these things together, or something else? And also, we don't well understand mechanism how these diseases grow and develop. On the other hand, the development of the drugs is very expensive and time-consuming. And for example, in 2018, pharma companies spent around $2.2 billion and up to 15 years to bring the one drug to market. But the bigger issue for pharma company than money and time is that then more than 90% of the new drug candidates never reach the market. So to better understand what is going on, we have to go to a pipeline of the new drug discovery. So everything starts in biological laboratories where the biologists and cell biologists perform experiment to understand the mechanism behind this specific disease on a cell and molecular level. Actually, they are trying to find the target molecule responsible for this specific disease. And when they identify that molecule, then the research moves to chemical laboratories where the researcher working to find a compound or compounds able to act on that target molecule. And after selection of that molecules, then the researcher performed a comprehensive physicochemical characterization of these compounds, and then the research move again to biological laboratories for evaluation the safety and efficiency of the selected compounds.
And if it's everything okay, then we go to a final stage, to clinical trials, where we evaluate the safety, efficiency, and long-term effect of these selected compounds on a human. So, as you can see, this process is time-consuming, but also is not so efficient. So, as you can see here, out of 10,000 selected molecules, just one reached the market. So this could be explained by the fact that the translation of the results from one stage to another is not so efficient, especially if you want to translate the results from the preclinical trials to clinical trials. Success is less than 10%. So why is this happened? Because the models that we use in the preclinical trials based on the 2D cell culture and animal models are not so efficient and adequate. So first, everything starts on the 2D models. Just briefly to explain, we use the cells, and then we seeded the cells on the flat surface in the plastic culture dishes. I stress the flat and the plastic. And then the cells grow and form the monolayers. And after that, we perform the different tests on these models. These tests are very simple, cheap, fast, repeatable, but the results that we obtain are not relevant and reliable. The reason for that is because the cells in our body don't grow in a 2D environment. They are surrounded with the extracellular matrix, and they interact with the extracellular matrix. So here you can see this is the cells, different cells, and these lines with the different colors represent different components of the extracellular matrix. The extracellular matrix play a very important role in the cell behavior through its structure and composition. And therefore, in a 2D cell culture, the lack of extracellular matrix resulted that the cells lose their morphology, of course, interaction with the extracellular matrix, and therefore they have a different metabolism and different responses to drugs. The animal models much better mimic what happened in the patients, but the test on these models first is a very complicated, time-consuming, always raise many ethical issues, and in the end, often produce misleading results due to the interspecies differences. How we can overcome all this limitation with this model? One of the possible solution is to create artificial in vivo-like environment based on the biomaterials as a cell carrier that mimic the native extracellular matrix. So the biomaterials or cell carriers that we use have to provide the adequate 3D structure, adequate composition, and adequate biomechanical properties, and the material that we use to produce the cell carriers have to be biocompatible. It means that material doesn't have any negative effects on cells. So when you create the cell carriers, you always have to keep in mind the composition and the structure of the extracellular matrix that you want to mimic. Generally, we can divide the cell carriers into two groups. So cell carriers for the soft tissue, such as ligament, blood vessels, or muscles, and for the hard tissue, such as the bone. So the extracellular matrix of the soft tissue are composed of organic components, such as the proteins, glycoproteins, and proteoglycans. And the best material to mimic the composition of the soft extracellular matrix is collagen, because the collagen is a major component of the extracellular matrix, or we can use silk or polysaccharides. And the form to mimic the structure is to use the hydrogels, or to use the fibrous meshes On the other hand, bone extracellular matrix is also composed of the organic part, but they also have mineral particles such as the hydroxyapatite. So here you can see this is the collagen fibers, and these green dots represent the mineral particles. So if you want to mimic the composition of the extracellular matrix of a bone, it is necessary that you have the organic components and also the mineral particles such as the hydroxyapatite, beta-TCP, or bioactive glass. And the structure suitable for bone cell carriers is to be spongy-like structure. So I will just move my talk to our lab to show you how we develop the cell carriers and how we use them for anti-cancer drug screenings. So we choose to work with the alginate. Alginate is a polysaccharide obtained from the brown algae. Maybe the alginate is not the best material because it's bio-inert. It means that alginate doesn't have any negative effects on cells. So the aqueous solution of the alginate in the presence of the divalent cations such as the calcium, zinc, or barium form biocompatible and biodegradable hydrogels. And we can produce a different shape of these hydrogels, but with a very easy procedure. And thanks to the hydrate, soft, and elastic structure of this hydrogel, alginate hydrogels well mimic the composition of the soft tissue, extracellular matrix of the soft tissue. Also, we can embed the mineral particles inside the alginate hydrogels, and in that way we can obtain the cell carrier for a bone tissue. So in our lab we developed one very user-friendly procedure for production of cell carrier. It means that this procedure can be used without specific equipment, but what is the most important, that every biologist can produce the cell carriers in our own lab without any technical expertise. So we produce the alginate hydrogels in the form of microfibers. You will see how we do that during our demonstration after a coffee break, but just briefly, in the syringe we mix the alginate hydrogels and the cells, and after that we extruded the solution in the gelling bath with the calcium ions, and we obtain the microfibers. So you can see here, this is the microfibers. All these black dots represent cells. And in this way, we can produce the kilometer and the kilometer of the alginate microfibers with the cells. And then we try to, in the same time, immobilize the cells and the mineral particles in alginate microfibers in order to mimic the composition of a bone, not the structure, just the composition. So we successfully produce this type of the microfibers. So the black colors of the microfibers is due to the presence of the mineral particles. And after extrusion, all these cells stay viable. So here you can see these green dots represent a viable cell, whilst the sporadically red dots represent dead cells. So these results indicate that the manual extrusion didn't have any negative effects on the cells. And then we wonder, can we use this model to cultivate the cells for short term, up to 5 days? Because this period is enough if you want to do the fast screening of your drugs. So we perform experiment on the different cell lines, but I will show you just one exam that we performed together with the PhD student for Premurosa in Italy. This is a part of Ella PhD. So we immobilize the human osteosarcoma cells in alginate microfibers, and we cultivate these cells up to 5 days, and we obtain that the cells, after 5 days of cultivation, the majority of the cell stay viable. And then we move one step further. So we check possibility to use this model for long-term cultivation, because this is important if you want to see the effects of your drugs in the long term. So we cultivate the glioblastoma cells in alginate microfibers up to 28 days. And we obtain, as you can see here, that the cell stay viable after cultivation. And this is, you can see this green line represent the number of the cells, so that during the cultivation, cells retain their possibility to proliferate in the 3D culture. And what was our aim is to use this model for anti-cancer drug screening. So we evaluate this model for rapid drug screening. So we treated the osteosarcoma cells immobilized in the alginate microfibers with and without the hydroxy apatite with doxorubicin. Doxorubicin is a drug of choice for treatment of patients with the osteosarcoma. And what we obtain that the cells in a 3D culture with and without the hydroxyapatite were more resistant to a drug. So this you can see that the concentration that kill half of the cells in your culture were 3 to 4 times higher than in a 2D culture. And then we checked what happened with the lung cancer cells treated with the cisplatin, also in the 2D and the 3D culture, and we obtained the similar results, but in this case, concentration was 25-fold higher in the 3D culture. So these results show that the 3D model better mimic cells response than the 2D culture. And then we check possibility to use this model to mimic to treat the cells in clinical relevant schedule. So we treated the glioblastoma cells immobilized in alginate microfibers with the temozolomide. Temozolomide is a drug of choice for treatment of patients with the glioblastoma. So the treatment lasted 3 days. And after that, we left the cells for the next 3 weeks to recover. On that way, we mimic the time between 2 chemotherapy cycles. So, as we expected, we obtained that the drug reduced cell growth. So the gray line represents the number of the cells in an untreated group, while the orange line represents in a treated group. And again, we obtained that the cells in a 2D culture were more sensitive to drug. And then we wonder, does it something happen with the genes responsible for drug resistance? So we checked the expression of the genes that code the proteins which pump out drugs from the cells. And we obtained that these genes weren't expressed in a 2D culture, so these gray bars represent the level of these genes in the untreated group, while the orange bars represent in the treated group. And you can see here that the level of these genes were significantly higher in a 3D culture. So these results explain why the cells in the 3D culture were more resistant to a drug. So just briefly to sum up, definitely to improve the translation of the results from the preclinical trials to clinical trials, it's necessary to develop relevant and reliable 3D models based on the biomaterials as cell carriers. We show here that we can use alginate to produce cell carriers for soft and also for the bone tissue, but you will hear later more about that during the event presentation. And we show that we can use the alginate microfibers for rapid drug screening, but what is the most important thing, we can use for evaluation of drugs in a clinical relevant schedule. So last but not the least, I want to thank to all the researchers who were and who are involved in this work, especially to girls from my lab and also for a team from University of Eastern Piedmont, led by Professor Lia Rimondini. So thank you for your attention.
Any questions?
Thank you so much for your presentation. It was really nice overview. I like the first part of, of your presentation because you really give us a strict message of what is important and why these models are important. I'm curious more on the part when you're doing upstream analysis for qPCR expression. We have some experience with alginate as well, but isolation, how to degrade alginate, sometimes it's so tricky. And RNA isolation, you do it with Trizol or with some kit?
Yeah, with Trizol.
Okay.
Because the concentration of the alginate is very low, it's just 1.5%, so we use the natrium citrate to dissolve. We first dissolve the hydrogels, and after
no
that...
enzymes. No enzymes.
No.
Nice.
Because you know how the alginated gel, so it's exchanged when you have a natrium alginate solution. When you put the calcium ions, then the ions exchange. And when you put the high concentration of the natrium again, then the ions exchange. So it's an easy way to dissolve. You will see later how we do that.
Yeah, okay. I'm curious because we have some kind of mixture of cell lyse with alginates, and we have some issues with isolation of RNA.
We have a problem when we put the hydroxyapatite inside to isolate the RNA. Yeah.
Okay. Thank you. Nice work.
Thank you.
Any more questions? Thank you, Jasmina, for guiding us to the reliable 3D drug testing, which will be also Jasmina, and later now it's joining Bojana. And Ivana will be part of demonstration here after coffee break at 11:20. So we invite you to join here at the table. And now next one is Bojana Obradović, also from the Faculty of Technology and Metallurgy from University of Belgrade. And she will introduce us what do biomimetic bioreactors mimic and why.
Okay, good morning. Thank you for the introduction. My name is Bojana Obradović. I'm coming from Faculty of Technology and Metallurgy and the same group as Jasmina, and I will talk today about biomimetic bioreactors that we work on in my group and also in the Premurosa project. And I will tell you a little bit what do biomimetic bioreactors actually imitate and why. So biomimetic bioreactors were developed for 3D cell cultures. As you just heard from Jasmina, so these cultures aim to establish environments so that the cells can interact with each other and also with the extracellular matrix in a 3D structure. And these cultures are important for drug testing, and as you heard just about the microfibers, but they were actually developed for tissue engineering. So tissue engineering starts with the idea of the tissue engineering is that we isolate the cells from the patient if he or she has some defect or disease, so that we can take a small biopsy, then to proliferate these cells and then to seed them on a scaffold together with some biochemical molecules, growth factors, hormones or so, and then to cultivate this cell scaffold construct in some cell culture, and then hopefully we will get the functional tissue sample that we can put back, implant back to the patient to the site of the defect. And if we think about cultivating this functional tissue equivalents, then we think, what do we need to provide to cells? If we just put this three-dimensional culture, the sample, in a static culture, then all these molecules and oxygen and other molecules are coming by diffusion through the scaffold. And diffusion can supply these biochemical molecules just to thicknesses enough to support the cells in 100 to 300 microns thick. So when we look at these microfibers that Yasmina just have shown, then you see basically here the green line green color shows the viable cells, so that all these cells, basically most of them, are really viable because these microfibers are 500 microns in diameter. So there are these distances that are enough for static culture and for diffusion. So there is no problem for that. But if we think about really samples of tissue, like if we want to make a cardiac patch, or if we want to treat the cartilage defect, or if we want to treat tendon defect, then there is no defect that is just on the size of microns. Basically, these defects are in sizes of several millimeters to several centimeters. So how can we then supply all these molecules to these samples so large? Let's see what is happening in vivo. In vivo, in organism, we have in all tissues and organs really branched network of capillaries that are providing with the blood flow all the necessary nutrients, oxygens, and then remove also metabolites. So the next idea, of course, is to provide also flow in our culture. So in this way, we can enhance mass transport. But if we again look at the static culture, where these cells are sitting statically in a petri dish or flask, and then we take a look what we do actually every day, that we walk, jump, run, and many other things in which basically all our tissues and organs are dynamically loaded and strained, stretched, and twisted. And even now, when you are listening to me, you are blinking, your eardrums are basically moving, receiving the sound waves, heart is beating, and then also we are breathing all. So basically, even if we are not moving, our all tissues and organs are in dynamic conditions and mechanically in some way strained. So basically, we need to provide a lot of different mechanical or physical signals to the cells. To provide this fluid flow and also these biophysical signals to the cell culture, actually we need especially bioreactors, biomimetic bioreactors, in which we cultivate these cell scaffold samples. So these bioreactors aim to imitate the environment that is in our body to cultivate specific tissue that we want. And basically, as our tissues in our body are really under different conditions, then we need to design biomimetic bioreactors that are basically imitating these particular environments. And as in Premurosa, we are working on focusing on musculoskeletal tissue regeneration. Then, and also we are working on those in my lab also. I will show you some examples of skeletal bioreactors for skeletal regeneration of skeletal tissues. And we will start with articular cartilage. Articular cartilage is a soft, vascular, this bluish-white tissue that lines bone ends in synovial joints. simply said, these joints are the ones that are moving in our body, like fingers, shoulder, wrists, knees, hips, and so on. So the function of articular cartilage is to transfer the loads in the joint. Here is a picture of the drawing of knee joint. So this is the articular cartilage, this is photograph, and this is arthroscopic image in the knee. So cartilage is transferring compressive loads in the joint to the subchondral bone that is below cartilage. And it withstands very high compressive stresses. They can be as high as 6 to 18 megapascals. For example, when you extend the hand and hold it like for several minutes, such 18 megapascals were measured in the shoulder of human. On the other hand, also cartilage provides almost movement of the joints almost without any friction. And when you look at the coefficients of friction, they are 0.005 to 0.04 measured in human hip and knee. And just to have the idea, when you slide ice on ice, the coefficient of friction is 0.01. So these ones are even lower than ice on ice. So it is really, from engineering point of view, this is amazing system. So this actually functions. Articular cartilage provides due to specific structure and composition, but we will now more concentrate on the different physical signals that we would like to imitate in the bioreactors and that are actually happening in the joints. So when you look at the knee joint, for example, here, then you can see when we are walking, then first cartilage is exposed to compression, so compressive loads. But then as it is moving, then we have also shear stresses from the fluid flow and from the basically friction between two surfaces. And then there is also tension as some surfaces basically stretched on the other side. And then finally, because we are compressing the fluid that is in the cartilage and in the joint, then there is also hydrostatic pressure. So this is quite complex, but usually key stress is taken to be dynamic compression. So in designing bioreactors, the first idea was actually to design bioreactors that are providing compression, dynamic compression to the samples. And here is actually one of these bioreactors that was developed in different labs with the same idea. So the idea is that you have this like chamber and then the wells in which you place the sample, and then on top you have this cover with these plungers, and this cover is then by a motor moving up and down, and it can be performed in this sinusoidal way and at up to 1 Hertz frequency and 5 to 10% compression strain. And what these several labs were they obtained was that this dynamic compression actually enhanced regeneration of the tissue, actually mechanical properties of the cultivated samples. But they also saw that it is really important how we apply in which regimes. So this is not yet discovered what would be the best, but especially since it really depends on how already developed this sample is. So if you have just cells in the scaffolds, then dynamic compression just is not good for them, just squeeze them. But what this also bioreactors revealed was that in this bioreactor we had again mass transfer limitations. So basically, because these samples were mostly covered by these these plungers on top, and just some mixing of the fluid inside, this was not enough to provide to this half a centimeter sample enough of the oxygen, especially, and the other molecules. So the next idea was to introduce in these types of bioreactors also fluid flow. And in my lab, we developed a bioreactor that incorporates tissue perfusion with dynamic compression. So basically, we have here a holder that holds 6 of these cartridges. So the idea is that you put the sample, you have the medium flow through this cartridge here, and then it is covered, the cartridge is covered by a diaphragm, and on top there are these micrometer screws that can be positioned to touch this diaphragm. So, and then all of this is then mounted on a base that is moving up and down, so that you can compress these samples. And also we have, of course, underneath the stepper motor that is moving this base, and also we have a load cell that is measuring the mechanical stresses on the loads that these samples are experiencing. So we can actually monitor the mechanical properties of the sample over time during the cultivation. So we use this bioreactor in some cell studies, but more in biomaterial characterization of novel biomaterials under physiological-like compression conditions. So we evaluated mechanical stability of the samples, where we could actually monitor how it is stable or not stable over time, and measure the dynamic compression modulus, Young's modulus also. Then we could also investigate cell biomaterial or tissue biomaterial interactions, cytotoxicity under compression, also bioactivity under compression. And here I cannot just not to mention a bioreactor that was developed in Davos in AO Research Institute, which is really for me very interesting, because it is even more physiologically imitating the environment in the synovial joints and here in the knee. And more you can of course ask Mauro Alini, who is here and invented this bioreactor. But just to show you, basically the idea is that here you have a sample, you have this ball that is basically compressing the sample, but then it is also rotating. So in this way you have this movement as in the knees when we actually move our knees. And so it is really physiologically relevant. Of course, when we go on into adding complexity to the bioreactors, then it gets more complicated to run the cultures. But of course, we aim to be as physiologically relevant as possible. And this bioreactor was used to actually investigate the regimes of the compression and this tension on the cells, cultivated cells, then to see the effects either of compression or tension and so on. And now I will move to bone. So bone is a completely different tissue. It is next to the cartilage, but it is highly vascularized, and it is also provides mechanical stability to the skeleton, and also provides locomotion and also protection of internal organs. It is it is self-regenerative, in contrast to articular cartilage, which has really low capacity for self-repair. But the problem is when we have large defects, and in healing of these defects when they cannot basically regrow. And in vivo, bones are submitted to a variety of stresses. So during our activities, we have tension, compression, torsion, shear, and so on. But what was interesting, in vitro, it was shown that for osteogenic differentiation and osteogenesis, the hydrodynamic shear stresses coming from fluid flow were the most important for osteogenic differentiation and osteogenesis. And where is this flow coming? Actually, as opposite in the cartilage, which is gelatinous, bone's structure is porous. So when you have like compression in walking or jumping, then this fluid is then forced to flow through these pores, and these cells, these dots, are basically experiencing this interstitial flow of the fluid, and they experience this stress. And this stress came to be very important in vitro, so when you want to grow bone in a lab. So of course, then the most obvious, the idea is to grow bones or samples that should be, that we would like to grow into bones in perfusion bioreactors. What does it mean? Just to have perfusion through the sample. And the most simple idea is to use just filter cartridges. And you can see here, just instead of a filter, you can put a scaffold. And you can do it in your lab, like anyone can make such a bioreactor. Of course, you can go on in a more sophisticated designs. For example, in the lab of Gordana Vunjak-Novaković, you have here like a perfusion bioreactor which has 6 valves, so that all these 6 places for the samples are perfused from the bottom up in this bioreactor in the same time. Our idea was to provide a perfusion bioreactor that is specifically aimed for the scientists without any technical expertise. So it is comes sterile and connected. So it's a single use. The chamber is silicone tubing which can be cut in the length that you want, and also we made it that you can use 3 sizes of the tubing. And we will show you, it is there on the table during the demonstrations. We use this bioreactor also within the Premurosa project for biomaterial characterization under perfusion, but also cell cultivation in macroporous scaffolds for bone tissue engineering and osteosarcoma research. And this will be the topic of and it's a topic of her thesis of Ivana Baničević, so she will show you this later after the break. So just to show you briefly, I hope I'm not completely over time. So how we combine both tissues, both concepts. So we have, if we want to cultivate osteochondral tissue, so very often these defects in the knees or in these joints are not only on the superficial layer of cartilage, but they go under to the subchondral bone. So if you want to treat this effect, then you would need to regenerate both cartilage and the bone. So you would need to have chondrogenic differentiation on top, like cartilage, and osteogenic on the bottom. And then you would need to provide dynamic compression to cartilage and a perfusion and vascularization for bone. And then these also cultivated samples, they should be mechanically strong if you want to imagine that you will put them in the patients. So the first idea in a lab of Gordana Vunjak-Novaković was to actually combine their perfusion bioreactor with this cover with the plungers that I showed you for cartilage. So in this way, to perfuse the samples and also to compress them. And also you need a bilayered scaffold, which is porous on the bottom and the hydrogel on top. So our idea was also to use our bioreactor for such purposes, and to basically we developed a new cartridge that has basically 2 inlets for 2 different media, and we have again porous bilayered scaffold made of gel and gum in collaboration with Sasha Novak and the Jožef Stefan Institute, and which is then porous, lyophilized in the bottom, and hydrogel is on top. And we tested this this scaffold in our bioreactor, and just to show you, this is how hydroxyapatite is formed in the lower layer. Hydroxyapatite is the component of bone. So this is the initial scanning electron micrograph, and this is under static conditions. Here you can see some needle-like crystals, which are hydroxyapatite in the static culture, but in the bioreactor culture you see that it is full of these crystals. So this is how you can use bioreactors for biomaterial characterization. But just not to finish on the simple example, I will show you now really advanced example from the literature from Gordana Vunjak-Novaković lab. They actually work a long time now on regeneration of temporomandibular joint condyle. This is this condyle, as you can see here, a bone with a cartilage in a jaw. So they did already 10 years preclinical studies in pigs in basically anatomically shaped scaffolds that they CT scan the defect, then they make the anatomically shaped this for each pig different one, different anatomy, so the shape, and then they make like a mirror image bioreactor chamber, and this chamber is then put in a bioreactor, and this part is for the bones, is perfused, you see, through many of these holes to have good perfusion, and on top is this chondrogenic media. So then they implanted these cultivated samples in pig's back, and so I think preclinical studies are now finished, so they will go very soon in clinics. So this is really, as much as I know, very close to one of the very rare examples that will go to clinical trials soon. So at the end, I would like just to summarize that biomimetic bioreactors aim to capture key physiological features of specific organ or tissue, and they have great potentials for different applications, for biomaterial characterization, for drug testing, tissue engineering, cell and tissue research, and also in designing strategies towards personalized medicine. They are in compliance with the 3 R's principle, which aims to reduce, replace, and refine the animal experimentation, as we have more reliable in vitro experiments, we would use less the experiments on animals. And finally, but to really use these bioreactors more widely and for different tissues and different of these applications, we really need a stronger multidisciplinary collaboration of engineers and life scientists. And finally, I would like to acknowledge my group in Belgrade. We are working on these bioreactors for different applications and also funding, including, of course, Premuroza project, and really great work done by Ivana Baničević that you will, with this perfusion bioreactor, that you will hear a bit later more about. And thank you for your attention.
How you deal with Gender diversity.
Gender diversity depends on, if you are thinking about personalized, tissue, you think gender diversity in my group. My group is really female group. We had some male students, but I don't know, maybe they just, they see many nice girls, as you can see, so maybe they are a bit frightened, but yes.
But you probably have cells from donors of different genders, right?
Yes, if we think about solutions for the cultivation, yes, this diversity, it is basically provided by, basically in our lab, we don't use human cells, we collaborate with other institutes, and then they have human cells from donors, and we use animal cell lines. In my lab, it's not for human cells. Yeah.
Your bioreactor for bone. How easy is to perfuse bone? Because what you show on the picture are micro, nano pore. It's the caniculi that go through the pressure, the fluid. But because many years ago, I know that Jeff started to develop a bioreactor, and we had a lot of problem of migrating the cell out from the bone, like fibroblast or preosteoblast or whatever. And that were a major issue because you get fibrotic tissue all around. You have the same issue, or you solve it?
Well, first of all, to say cartilage doesn't like actually flow. This is the problem in these bioreactors growing cartilaginous tissues, because you have negative effects of the flow that is making fibrotic capsule, and on the other hand, if you don't have flow, then you have not enough regeneration. So this has to be basically optimized. So in our bioreactor with dynamic compression for cartilage, we had just some not big pieces of cartilage. So because of this issue, we used it more for biomaterials characterization in collaboration with different groups. For perfusion bioreactor that your question is related to, this bioreactor would be suitable for really biomaterials. And if what you will see in even a stock, when you seed the cells inside and then you perfuse, and this is macroporous, not nanoporous, but macroporous scaffolds, where you can have a good flow and not high pressure drops, and not the pump is just peristaltic pump and it's easy. So you have to have really porous scaffolds. When it comes, we are thinking about establishing ex vivo, basically explant cultivation, and with the bones, and this seems that it will be a problem, exactly what you said, but this is a project of the next student. So we hope to come up with some solutions, maybe very thin samples that we could perfuse, or just not to really completely perfuse them. We will see how it goes, and maybe we talk about experiences, because the bone would be really, it's already stiff structure. So
You should only try at the beginning with stem cells.
with cancellous bone. Yeah, yeah.
Cortical bone.
Not cortical bone. Yes, yes.
I know it's halfway through
Yeah.
the real bone, but
Yeah, but still, yeah, I think this will be something like that, yeah, because this bioreactor really doesn't allow for high pressures. I mean, it's all like silicone tubing. It will just, you know, expand to allow bone to go, to allow flow to go around. So yes.
Thank you, Bojana, for a very interesting talk. And now we have short coffee break. We will see it's outside. And we then continue with, at 11 with Ivana Banicevic. After that, we have the practical demonstration of the perfusion bioreactors Bojana mentioned. And at 12, there is a roundtable on precision medicine, how far we are. Hello, welcome back. We are moving on with our program. Now with us is Ivana Banicevic from Faculty of Technology and
So, as you could already hear, we are facing still some challenges such as organ shortage, unreliable drug testing, and so on. And we are tackling these challenges by using 3D in vitro culture models. These models are combining cells, biomaterials, and bioreactors together so we can imitate more closely human tissues. These models are highly used in tumor and tissue engineering. Okay, so based on this, our idea was to develop 3D in vitro culture model that we can use to imitate both healthy and diseased bone conditions. Concerning diseased bone conditions, we were particularly interested in bone tumor osteosarcoma. Before I proceed with showing you our model in more details, I think we should first understand why bones are an important focus of 3D culture models. So bones, and you heard from the previous lecture a lot about bones, but you know, bones are much more than rigid structures in our bodies. They have some obvious functions and some less obvious functions. They have very interesting and unique composition because bones are made from extracellular matrix that is composed of mineralized tissue, and that gives bone stiffness and strength. However, bones are not heavy, they are lightweight because of the porous structure, the one you can see in this photo. Bones are prone to many diseases. One of them is osteoporosis, which usually affects older people. This disease is actually decrease in bone density, which makes bone weaker and more prone to injuries. One of the most common type of injuries are bone fractures, and unfortunately bone is common site for tumor development. Concerning bone fractures, bone has capacity for self-regeneration, but sometimes those defects are too large to heal and surgery is needed. So that surgery actually means to bridge the gap that is actually defect with another piece of bone, and that piece of bone can be from the same patient, from another human, or from an animal. The major challenge here is donor and organ shortage, and it would be really helpful to have bioengineered bone grafts that are personalized and that can be used for this challenge. Another bone disease are bone tumors, and bone tumors actually introduces an entirely new set of hurdles. Primary bone tumors are the one that originally takes place in bones, and they are hard to treat. Also, no major improvement in therapy in the last 20 to 30 years. Again, you heard a lot about this from Yasmina's lecture. Here I would just like to state that bioengineered tumors could actually improve drug testing because they are more alike human tissues. Therefore, the process of drug discovery would be much more efficient. Both bone tissue and bone tumor engineering relies on the use of 3D culture models. So now I'm back to our 3D culture models. The choice of cells in our model depends on the condition that we imitate. If we want to imitate osteosarcoma, then we grow osteosarcoma cells. But if we want to imitate healthy bone, then we grow mesenchymal stem cells. Mesenchymal stem cells are versatile cells that can turn into different cell types, including cartilage, bone, fat cells. And it is quite interesting to explore what conditions are needed for mesenchymal stem cells to specifically and directly turn into bone cells. Another component of our model are scaffolds. So we make our scaffold from alginate, hydrogel, and hydroxyapatite particles. alginate hydrogel represent organic phase of our scaffold, of actually bone, and hydroxyapatite particles represent mineral phase of the bone. With this composition, we aim to imitate actually bone composition. Now you can see the scaffold, and you can see scaffold, you can see a scaffold cross section, where you can see that scaffolds are spongy, they have pores. Because not only we want to imitate scaffold composition, we also want to imitate bone structure. So now I'd like to go through the scaffold production, and just briefly. So we start by mixing two main components, alginate and hydroxyapatite. We get suspension that we pour into dialysis tubings, which serve as a mold and to keep our suspension in one place. Then we put that dialysis tubings into solution for alginate gelation that is based on calcium ions, because calcium ions turn alginate solution into hydrogel. After gelation is finished, we have these compact cylinders which we freeze. Freezing is followed by freeze drying, and these two last steps actually are important for creating pores in our scaffold. After freeze drying, our cylinders are completely dry. We rehydrate them a little bit, then cut them into discs, and then we obtain scaffolds that we use in our cell cultures. When we have our scaffold ready, then we can seed cells manually onto the scaffold. We cultivate cells for 1 or 3 days in a dish under static condition to let the cells attach better to the scaffold. Then we place this scaffold with cells inside into perfusion bioreactor chamber, where we cultivate cells for 7 days. So here you can see in this photo perfusion bioreactor, and conditions were completely the same for both osteosarcoma and mesenchymal stem cells. In this perfusion bioreactor chamber, as again you heard a lot about this, there is continuous medium flow throughout the scaffold with cells, so there is constant supply of nutrients to the cells, and also shear stresses are acting on cells. As control, we use scaffold with cells under static conditions, and in that way, by comparing perfusion and static conditions, we can clearly see what are the effects of perfusion conditions. Okay, now I'm moving to some of the results that we have. So first thing that we check always is cell metabolic activity. It gives us nice information about how the cells are in our model. So for osteosarcoma model, we performed MTT assay. This assay actually turns metabolically active cells into these black crystals. And here you can see these scaffolds are stained in black, which means that they were full of metabolically active cells. So then we dissolve these crystals and we measure absorbencies of solutions. Higher absorbance means higher metabolic activity. So from this graph, you can see that cells had highest metabolic activity in perfusion bioreactor, which means that they benefited from increased mass transport. We also checked metabolic activity for healthy bone model, for which mesenchymal stem cells were used as representatives. And here in this graph, you can see that metabolic activity of the cells is quite similar. So we have two different results for two different cell types, and we can explain this by taking into account that osteosarcoma cells are actually tumor cells, and that they in general have higher metabolic activity, so they will be more sensitive when there is limitation in mass transport. That is, they will benefit more when that mass transport is increased, as it is the case in perfusion bioreactor. Okay, so another thing we wanted to check for osteosarcoma model is their shape and how they are attached to the scaffold. For that, we did scanning electron microscopy. So these are the micrographs, and you can see that the cells are round, have round shape, especially after seeding and in bioreactor, and they are attached to the scaffold. Also, you can see that they are in the form of aggregates. So in order to visualize where are the cells in our scaffold, we performed histopathological stainings on scaffold sections. First of the staining is hematoxylin and eosin staining, which stains alive cells. So here, in this micrograph above, you can see the whole section of the scaffold, while down at the bottom is the section in higher magnification. So these dots are actually alive cells within a pore, and you can see that they are close to each other, but still they are in the form of loose aggregates. So after short-term cultivation in bioreactors, cells, as you can see here at the bottom, grouped and formed these very compact cell aggregates, which is really characteristic of tumor cells. Also, in static, we notice that cells group and form these aggregates. We measure the size of aggregates in perfusion and static cultures, and these aggregates had a larger size in bioreactor. Another thing we did was reticulin staining. So reticulin staining actually stains collagen fibers that are secreted by osteosarcoma cells. Again, from this micrograph, you can see that cells grouped. But in higher magnification, you can see that osteosarcoma cells in bioreactors secreted more of those collagen fibers that are pointed with yellow arrows, while in static, these fibers are not so much present. So it seems that perfusion bioreactors actually encourage cells to secrete more of those fibers in perfusion bioreactor, and to secrete more of extracellular matrix. For healthy bone model, we performed some of the same analysis. For example, hematoxylin and eosin staining. Here you can again see how the cells are distributed in our scaffold. And here you can see that the cells didn't have tendency to group in these spheres and that kind of aggregates. Rather, they form these aligned structures along the pores, especially under perfusion conditions. The same you can see from scanning electron micrographs. You can see that the cells, it seems that the cells grew in one direction in bioreactor, probably because of the flow presence. Also here you can notice that cells had elongated shape, so they retain their original shape, in contrast to osteosarcoma, which are round in our within our scaffolds. Also, for this healthy bone model, we checked we did F-actin staining for mesenchymal stem cells, and you can see here this F-actin is actually that is protein that is inside the cell. It is main component of cell cytoskeleton, and it is stained in red in these micrographs at the bottom, while blue are cell nuclei. You can see that in bioreactor, cells have elongated shape. They grew in this very regular formation that looks like fibers, that is, or bundles, and in static they were more spatially disorganized. So, to conclude, for osteosarcoma model, we saw that osteosarcoma cells in our model show some typical tumor cell behavior. They had high metabolic activity. Also, they grouped in these aggregates. And I didn't show you here, but we checked some genes, and we saw that the cells retained tumor-related genes in our model. And the next step is to test anti-cancer drug in this model. For healthy bone, we saw that our model can support these mesenchymal stem cells and that perfusion conditions, which we found interesting, are inducing their spatial arrangement. Still, we need to optimize conditions for their cultivation. So with this, I would like to finish and to thank the Premurosa project that allowed me to work on this in this topic and to collaborate with all these institutions in Serbia and abroad. And many thanks also my main supervisors from the Faculty of Technology and Metallurgy, Professor Bojana and Jasmina, who guided this work.
from the reservoir, you can take medium out and put a new one, debubbling from these two syringes, and basically this is the system. Yeah.
So this setup that I see on the table, this is basically everything that I would need if I wanted to, let's say, create brain organoids in my garage, or is there something else involved?
Well, yeah, that is the only thing, except for you would need to have a sterile laminar hood. You would need to have cells.
As far as
yes,
scaffolding and
scaffolding,
pump.
you would, yes, the scaffold you would need to adjust or design a scaffold towards that tissue that you are planning. This scaffold is with these hydroxyapatite particles or bioactive glass particles and with alginate, so it's meant to be polymer mineral, so to imitate bones. Brain, you wouldn't have the mineral phase, but and of course I'm not sure for brain, or again, each tissue should check if it really likes perfusion, how much of this perfusion and in which places this perfusion would go. Do you want to make channels, or the flow could go around, or what would be? But this is basic setup. Also, what we also put in this bioreactor is these alginate microfibers with that you can test the drugs. So short-term drug testing, for example, in this bioreactor is completely simple. I mean, it's not as I showed you. I didn't want to show you just simple solutions. I showed you some also complicated ones from the literature, and also from our other bioreactor is more complicated. But this one is for perfusion is simple, and we tried really to simplify it for use.
And the temperature and CO2 regulation,
this is just put in the incubator. So incubator regulates everything. So we don't have a system for. Then it would be more expensive and complicated. And the idea is to be just simple. And we can demonstrate this. We can demonstrate alginate microfibers. So how to make them? It's easy. So this is the alginate solution. I just need a needle. So the diameter depend of the diameter of the needle, depend the diameter of your microfibers. So, and this is the solution with the calcium ions. So if you want to produce the fibers, then it's necessary to put the tip of your needle inside the hydrogel, inside the solution, and then something happened. I'm not sure that you can see here the fibers, but I can come to you to see, so most of you know how it looks like. Okay. It's like spaghetti. So, but if you want to make microbeads, so you can just put the syringe and the needle above, and just so inside is the.
and for encapsulation.
Yeah. So if you want to imitate the spheroids for cancer research, you can produce the microbeads, but they are bigger. So then the other methods we can use. So just see. So this is microfibers that we produce on this. Uh-oh. So, okay. And in that way you can put inside the cells. So this is a very simple method for cell localization. And of course, if you want to dissolve, then you use some solution with a high concentration of the sodium. Do we have? No. We don't have a solution to dissolve the microfibers, but this is a procedure. So on that way, we use this procedure to immobilize some active substance, for example, the silver nanoparticles, because for a long time we work with the silver nanoparticles. We develop procedure to produce the silver nanoparticles, and after that we immobilize in the alginate microfibers with intent for to be the wound dressing. So you can see how these fibers look like after we dry them. So it's like wound dressings. So this is alginate microfibers with the silver nanoparticles. It's brown because silver nanoparticles are brown basically when you produce them. So you can touch. You can use microfibers like a dressing, or you can also produce powders as these beads that we showed, or here we have also the alginate microbeads with the activated charcoal. Again, the idea is for removal of toxins from the wounds, and this is also we can have bioactive substances on the charcoal that is then for example, iodine that is killing bacteria, so fighting infections in the wounds. So we also just brought this to show you it's not related to Premurosa, but we worked also on these materials. And we can also produce with silver nanoparticles also different those films with silver nanoparticles that are practically soluble in the wound, or they could be insoluble in the wound, just absorbing the fluids. And but basically it's all based on alginate, and we really have long time worked with alginates and in different shapes, different characteristics, and it's not really any chemically modified or something, which also could be done in different laboratories. They do it to be more stable, more rigid, or more active, like attracting cells. We do just simple things, but it could be with the same simple approach, could be also improved, let's say, or make it more complex. Yeah, you can, well, usually people come to our table and then they look, especially kids like to come, we always show them how we make microfibers. Now, you can take a look at that, and Farah from the University of Eastern Piedmont and Leah's group will show her approach and the bioreactor.
So today you've heard a lot the word perfusion, right? So what we care about is to allow the food to enter into the scaffold so that cells get nourished, but also to let wastes go away, because if wastes accumulate, they're going to kill cells. Perfusion can be of two different types. The bioreactor that Professor Bojana, Ivana, and Yasmina has just explained to you about, you can see that the scaffold is within the solution. So it means that the media is passing within the scaffold, but also around the scaffold. This is called indirect perfusion. In our lab, we actually work with direct perfusion. What does it mean? It means that we want the media to be passing only through the pores of the scaffold. For this to be possible, we need to be very precise when it comes to the measures of everything, and that's why we need everything to be 3D printed. What we work with, they are scaffolds that are 3D printed to have the diameter exactly of this holder. Usually we work with diameters that are around 1 centimeter and a thickness that is around 0.5 centimeters. Then this scaffold that is 3D printed, we place in this holder. This holder will then be placed within that chamber that was also 3D printed to fit exactly this holder. Everything will then be closed. What happens is that the media will be moving down and going up, but since the only space that the media has is the pores, or they are the pores of the scaffold, so media will just be passing there. This means that we will be achieving direct perfusion. We added also a new element to the bioreactor via our collaborators, because as you also saw today that we try to replicate the environment of a tissue. We try to be as close as possible. We're never 100% replicating what happens in the body, but we try to be as close as possible. So that's why now our bioreactor does not only give us direct perfusion, but it also gives us hydrostatic pressure that can easily be applied simply by just blocking the fluid. So the fluid is passing, you block it. This blockage of the tube will cause a pressure caused by the liquid. That's why it's hydrostatic pressure to be applied. One important thing to mention is that, as Professor Bojana suggested, that you can change the settings. And this is very important when you work with bone, because you can be at rest, but you can also be in movement. So the perfusion will be different, the pressure will be different in both states. That's why when we do our experiments, we make sure to imitate rest or physical movement. I am also done, and if you want, you can also try to build the chamber yourselves. Here, unfortunately, I don't have the reservoir of our bioreactor, but what happens is that these two tubes are usually connected to a bottle. This bottle has media, and the media passes from one side to the other. You're free to build everything.
Basically the same pump you can use, so it's the
exactly
same tubing, it's for pumps, so basically it's just different producers and different sizes, but it's the same idea, yeah. So just yeah, you can come.
Yeah, exactly.
Yeah,
it doesn't
really
happen all the time to have the chance to touch what we work with in the lab.
One question, how can you control hydrostatic pressure? Like, do you know exact value, or you have some range of values when you press the tubing?
Because we have a pump, a pinch valve that you can moderate via a system, and you moderate it in two different ways: the time of the cycle and how long the pinch valve is closed. Then there are pressure sensors, and they are able to understand, okay, if I apply a cycle of 10 seconds, 5 seconds it is closed, 5 seconds it is open, it refers to a certain pressure that is within the physiological range.
Because honey is also
decrease the pH of collagen, because they cannot do this at the right condition. We are sure once we have pressure sensors, these settings will change, because you will have
yeah, I would like to listen to pressure sensors, but yeah, this is like we also have a moment. The system where you should measure things, and then the things that is there that you hope it will stay.
Yeah, exactly. Exactly. So here you will have the scaffold inside this holder, okay? And you can decide also how you want the flow of media to be. So do you want it to go from the bottom where the scaffold is, or you want it to come from the top? You can also decide. The most important difference, to be honest, just it will affect the orientation probably of cells. So, but anyways, the final result will be the same because the direction of the flow is always like this, so straight.
What is the material that you use for 3D printing?
We definitely don't use this fragile material. We use polylactic acid because it's very hard. So the material is very hard. If you touch this, you can notice it's soft. And definitely, this soft material, while being in culture, their properties change. So I'm not sure that still they are fitting in this diameter. They can swell, they can degrade. Polylactic acid stays the same. So the moment I put the scaffold until the moment I remove it, and we work with long periods of time, so up to 21 days. So after 21 days, the scaffold is still the same. I collect cells and do the studies on cells, or while the cells are in the scaffold, we image them, etc.
Do you have to 3D print for every experiment, or you can sterilize and reuse?
No, for every experiment, yeah, we don't reuse the scaffolds, especially for RNA extraction, because you degrade everything. So,
okay.
Yeah.
So this is 3D printed?
Yes, everything is 3D. I know this is PDMS. This, I'm sorry, I don't know. But one more thing that would be interesting also for you to know is that when we want to 3D print materials, there are also different types, and we should always make sure that what we are 3D printing is biological grade. So what it means is that these, every time I need to do an experiment, they should be sterile. That's why I need to make sure that this material can be autoclaved, so can undertake very high temperatures for it to be sterilized, okay? Because these are always reused. You just need to, even the tubings, we sterilize everything. So everything should be of biological grade, let's say.
if you can exchange between you, that would be perfect. Yeah, so if you feel free, please, to answer any questions you would like. Just let me know, and so I have some specific ones for you. But if you feel that you want to,
So we just signal you.
Exactly. Thank you so much. Okay.
Welcome back from the demonstration. Now we're starting with the round table, precision medicine, how far are we? And the round table will be moderated by Petra Prešeren, who will also introduce the speakers and the topic. Enjoy.
Thank you so much, Sasha. Welcome, dear guests, here and online, and of course, dear panelists. It is my pleasure to welcome you to today's roundtable discussion, where we will be exploring a critical and timely topic, the advancements and the future of precision medicine. We are particularly excited to present the Premurosa project, an initiative that aligns closely with the pursuit of personalized healthcare solutions. As we have heard in some of today's presentations, the Premurosa project is at the forefront of integrating precision medicine with compassionate, patient-centered care. Premurosa project focuses specifically on precision medicine for musculoskeletal regeneration and prosthetics. It aims to bridge the gap between cutting-edge scientific research and real-world impact on patients' lives. I believe all of us hope to live long lives, envisioning a future where we remain healthy and enjoy a fulfilling lifestyle as we age. However, the reality can be quite different. Musculoskeletal diseases, which are often associated with pain and limited mobility, are reported to affect roughly half of the world's population over 60. These conditions affect muscles, bones, joints, and surrounding connective tissue, significantly impacting the quality of life. With population growth and aging society, the number of people affected by these conditions is rising rapidly. And this is why the Premurosa project is so crucial. By focusing on optimizing treatments, reducing side effects, and ultimately enhancing patient outcomes, Premurosa aims to address these challenges and to improve the lives of those affected. Today, we are joined by a distinguished panel of experts who bring diverse perspectives and deep expertise in the field of precision medicine. Together, we will explore how far we have come in this exciting field, challenges that remain, and how projects like Premurosa can contribute to the shaping of the future of healthcare. Thank you all for being here, and now let's turn to our expert panel to begin this interesting discussion. In my introduction, I mentioned the terms precision and personalized medicine, and maybe this has caused some confusion. To give a better understanding of these two terms, I would like to ask Professor Dr. Mauro Alini for some further clarification on what is precision and what is personalized medicine. Professor Alini was the Vice Director and the Head of Musculoskeletal Regeneration Program at the IO Foundation, which is a medically guided global network of surgeons. It is the world's leading education and research organization for trauma and musculoskeletal treatment. Since last August, he is also an emeritus research advisor at AO. Professor, please, can you give us some examples and further clarification?
Thanks, Petra. I'm only an emeritus since last August. Precision and personalized words are similar. I would say that personalized medicine was old, the first definition. Precision is the new. Mainly precision means that people are looking to understand or discover the genomic of the patient or associate disease, the environmental, and the lifestyle. So all these three components will help to understand why a disease develop, and how better is the or how the best treatment will be assigned to the different type of disease. So personalized and precision can be used both, and the meaning is the same. I think it's important to understand that precise medicine, it's mainly driven to a type of tumor or a type of disease, to understand what's the changes at the genome level, lifestyle, what you eat, and your environment. It's obviously a lot of data that you collect, and you have to analyze to make different correlations. So that's why big data analysis, computational, artificial intelligence are important technology to be used in order to make sense of all these big data that you are collecting.
Can I ask you further? Does this mean that precision medicine, that a specific medicine or treatment is designed for a specific patient? You said that you're taking into consideration environment, genomics, and so on. So is this for each and every person or for groups of people?
I think, both can happen. Rare disease, where you have, for example, a few mutations that the consequence of these few mutations are, for example, osteogenesis imperfecta, which is a bone disease. It's mainly personalized because you have maybe few people. Cancer, breast tumor, you have 20,000 or 100,000 patients, and then the personalized is almost impossible. But the precise medicine will help you to at least select or differentiate between different breast tumor or osteosarcoma, if we want to stay on the musculoskeletal. That will help to improve the drug treatment. And that includes also, for example, biomarker. In addition, biomarkers are molecule, peptide, or that are present in your blood, or urine, or in liquid of your body, that can help to have a better diagnostic of what's happened, which type of tumor you have.
As you mentioned, precision medicine has gained significant attention in recent years and is already being applied in areas like oncology. However, in orthopedics, its application remains largely confined to adapting device geometry to match the patient's anatomy. Professor Dr. Abi Pandit, you are the president of the Global Tissue Engineering Regenerative Medicine International Society, which has a global mission to advance tissue engineering and regenerative medicine worldwide, to generate knowledge, to improve patient outcomes globally. You are also the director of the Center for Research in Medical Devices, based at University of Galway, Ireland. Can you please tell us how far we have advanced in integrating precision medicine into the field of orthopedics, and what are the next steps?
A very good question, and I think the first piece I think Professor Alini alluded to is the realization that there is a gap in the field. One is that we do need that personalization, and the reason is very simple, because if you look at how the studies have been conducted in the past, what kind of population have these studies been conducted? Have we looked at gender differences? Have we looked at difference in population stratification X, Y, and Z? It has not been done. So implants have been made for a particular population that have been studied. And that is a limitation in the field of medical devices per se, or even for drugs per se. I mean, so there is only a particular population. So the idea that this has to happen is the first step in any field. So that's a good thing. And you're right that personalization has only happened in the context of shape. And we can print 3D printing plates for fractures and X, Y, and Z, and they have made to the clinic. And there are beautiful stories, success stories in the field associated with that. So at a macro level, yes. To the point of acceptance of that implant, if I were to say in the medical device field, that has not happened yet. Because we haven't really understood how the host is integrating that substrate. Because I'm different, you're different, I'm of a different color, you're a different gender, X, Y, and Z. There's so many different age, so many dynamics at play. We haven't really understood those molecular mechanisms that are present. So the field has realized that that's the gap, and that's what most of the investigators are working on in the context of how can we address this issue? How can we understand if I use the word crosstalk between different cells, different disease states, and come up with solutions thereof. So the acknowledgement that it is something that we need to look at is there. We have the tools, and from cancer biology and other omics tools, we have a lot of tools present. So there are tools. We need a more concentrated effort to make it happen. The industry also has to realize it. Industry needs to invest in it, and the regulators also have to understand that this is what's needed.
So there's quite a lot of work ahead to check all of these aspects that you mentioned.
Yes.
Maybe Professor Padint or Professor Alini, can you tell us what are the advantages of precision medicine in advancing musculoskeletal regeneration? You have already mentioned some of the things, but maybe can you be more specific about it?
So as Professor Pandi already mentioned, we have a big gap. And this gap needs to be filled if we want to really catch up, for example, compared to tumor, to cancer research. Because we don't know what are the environmental and lifestyle influence on musculoskeletal. We only know the shape. So because we do an MRI, okay, this is the gap, 3D printing you can, but that's it. So, and that's a major concern. There are steps or fields that are improving. I can mention, for example, arthritis rheumatoid, where now they start to stratify patients also a little bit bottom up instead that top down. Bottom up means that there are patients that do not react or respond to drugs. Because there are 2 or 3 drugs in arthritis rheumatoid, methotrexate, antibody, and a few other, and we know that 1/3 of patients only react and the other do not. So they are trying to select which patient react to the drugs in advance in order to start with the appropriate therapy. But that is, it's a bottom up rather than understand the genomic and the complexity of the patient and the disease in order to come up with the best treatment. I don't know if Professor Ebi?
Yeah, no, I agree with you. I mean, I think we need to really, really understand the mechanisms underneath. Why are we getting a response? So, I mean, a classic one is one of the biggest failures in the field was on wear debris. Why do some specific patients failed, and why they didn't? There was a big, just for the audience, there was a big pullback on some of the wear debris that was occurring in orthopedic implants, and mainly weight-bearing implants. And the mechanism thereof, there have been many postulated one, but not all patients responded the same way. So, and why is that? And maybe if we understood that's the population that we should not be implanting X, Y, and Z, we wouldn't have done it.
So thank you for this short introduction, and I think now it's time to face the Premurosa project. I already mentioned some of the key objectives of the project, but I would like to go more in depth, and of course, who better to ask than the coordinator of the project, Professor Lia Rimondini from the University of Eastern Piedmont that coordinates the project and is a prominent academic and researcher. She has a degree in dentistry and is a full professor of applied medical sciences and technology. Professor, can you tell us more about the goal of the project that started 4 years ago?
Thanks, Peter. Yes, the goal. So the starting point were well underlined by Professor Pandit and Professor Alini. The problem is that we have a lot of materials with good results, with different chemistry, with different surfaces, with different mechanical properties. We have a patient from the other side with the biological responses and a different way to respond to biomaterials. And in the middle, we don't know what's happened till now, because we had no technologies available to study in deep the biological response to biomaterial as well as in other conditions. What we have now, that we have technology to get a lot of data, and now we don't know how to do with this data. So that is this means an Esopo tale, no, in which we have no food before, we have food, but we have no way to eat it. So the goal of Premurosa was to match the biomaterials and their properties were characterized, and the patient and their response were characterized, and to develop in between the tools for do it. That means new in vitro assays, more closer to clinical condition, with not the bias of the animal studies, with taking account what we can do in reproducing lifestyle and so on. We know now, for instance, that immunoreaction is gender dependent, and the way to react with inflammation or pro-regenerative effect depends on sex, for instance, as well as depends on age. And so what we try to do is to try to fill some gaps that remain huge. Talking about Premurosa, that it's interesting, we were able to convince the European Commission to grant us. And 4 years ago, we received about 3.5 million for the project. The project has a nice strata because it's composed of 13 sub-projects, each of them led by a young scientist. And now we obtain a nice good result because we were able to develop some in vitro assays with 3D cultures, with co-cultures, new materials. We even propose some new taste for standardization. We just published a nice paper in this regards. We introduce a strong part with a mathematical approach to manage the data. This is a very, very trendy scientific approach, but we believe the integration of computational part is very important to understand something more about the response to the body, to disease and so on.
And maybe you can say what the Premurosa means.
Ah, yes. Premurosa is an Italian word. It means so to take care with care, just to be very careful to all aspects. And so it was invented, I remind well, during the dinner with Mauro and some other young scientists of EU Foundation, and with a, I'm sorry, a good bottle of wine. We that is, but I remember also the bottle. Yeah.
So good company with experts and full disclosure about the wine,
Always,
okay.
always.
And
yes.
I just would like to add that we also develop NGS, new genomic sequence. It's not that. New generation scientist, which is extremely important because, as you see, we are. I am already phasing out. The author will do it soon here, and we need at least 13 new scientists that continue to work on this issue of musculoskeletal regeneration.
And as we heard in today's presentations, you did a great job. You have nothing to worry about the transfer of knowledge to the next generation because it's very impressive what they did.
Yeah.
Professor
So,
Pandit?
I mean, I think I agree with what Professor Alini has said. I mean, our success, I mean, we may not get a product out in the market. Most of us, actually, academics don't. 99% don't. But our best products are the other graduates which we turn. And the innovation that they do, wherever they go, whether they go in academia and industry, if they go in academia, they'll train out more of these products, or if they go in industry, they have the tools for innovation to do. So, you know, so thanks to Leah and her brainchild to create this, you know, this abundant of knowledge to put into this project is fantastic. And let's not forget, this was also done in COVID times. So it was very difficult at first to engage, but nevertheless, you see the results that we see is fantastic.
Thank you so much, Professor Pandit. And you already mentioned some challenges like COVID, but I would like to go again more in depth. What kind of challenges did you face during the Premurosa project? And Professor Dr. Bojana Obradović from the Faculty of Technology and Metallurgy, University of Belgrade, Serbia, has a doctoral degree in chemical engineering and is a renowned expert in the field of biomedical engineering and tissue engineering. Please, Professor Obradović, can you elaborate a little bit more on the challenges?
Yes, I would agree. First challenge was really this COVID, which prevented actually 1 year of meeting in person with the whole group. And luckily after that, when we had finally the whole group meeting, and it was in Belgrade in summer school, then it was real fun to meet all the people, professors, mentors, and 13 really bright young scientists together. And after that, really the Premurosa took off with activities which were really fruitful and beneficial, and very beneficial for students. I would say for extremely, as from our experience from Ivana, with really interacting and exchanging ideas and working with other students and also being friends with them, it really means a lot. So this was really, the beginning was really challenging. And for the second challenge, I would just say that anyone who is working with cells knows what is the main challenge. And this is working with cells. Because cells, as I am a chemical engineer, I worked also in studies of chemical engineering with chemical substances, with some, again, chemical reactors, not bioreactors. And there, what you put, you mainly can predict, of course, if you know chemical like mechanisms or what, anyway, what you are investigating. In the field of cell cultures, cells are living organisms, so they can change. They can respond in completely unexpected ways. They can be contaminated, so you work 3 weeks or 4 weeks and then you throw away everything and then you start again. And yes, working with anyone who goes into this field has to be aware that these experiments last longer, they are more challenging, they need effort. As I said, you cannot put the bioreactor on and it's working. In chemical reactor, probably it would be okay after 2 days, because if you know what you are doing, of course. But in a bioreactor where the cells are growing and may be contaminated, or CO2 may be built up, or whatever could be leaks, it's completely different story.
So you learn something new every day, you and your students, I guess.
Definitely, yeah, a lot, yeah.
You already mentioned before that not all research manages to get to the point from being translated from innovation into clinical practice. And Professor Dr. Michael Gessig, you come from the Aalto University Foundation in Finland. You're the European Orthopedics Research Society Ambassador for Finland, and you have 2 doctoral degrees in materials technology and composite materials, and over 25 years of experience in research and development of biomaterials. Is this one of the biggest challenges, translating innovations into clinical practice?
Thank you, Petra. I think that what has been mentioned by also Professor Pandit and Professor Alini is that not all the research development in the biomaterials, whatever field is going to clinical practice. And even if we know the mechanism, even if we know everything, how it works and why it doesn't work, there is another issue, because the healthcare medical industry, especially medical devices as well, they're highly regulated. That means that we have to also, beside functionality, that this material works, or we know that it works in such patients, we also have to think about safety. So, and when we talk about musculoskeletal application, the safety is it's not like drug which you eat, today, and maybe tomorrow you have some dizziness, but then after tomorrow is okay. We talk about 10, 15 years, 20 years, maybe more, especially for young patients. And we have very limited possibility, if at all, to know how particular biomaterial or implant, whatever it's metal, bioglass, tissue-engineered product, will work in 5 years, in 10 years, in tomorrow, what comes the infection, as we have this morning, or what comes with inflammation, how the body will react. And all these questions will be asked by regulators, because they want to know that when they give you a right to go to make available for the market, to give CE mark in Europe or FDA approval in US, you need to work with the safety. And that is one of the issues which is the regulatory science, which I think is one of the gaps what we discuss in Primorosa, and we also try to address it as much as we could.
It's one of the problems, I guess, because it's very expensive to do all this, but also because it takes such a long time to test for everything, right?
Well, I think it's everything, because you need a lot of time, you need a lot of resources to do that, and you have to know what you do, because that's another challenge. Because if you don't know what you do, that you have to convince other people and regulators in EUDAMED or in EMA to say that this is what the results, what you show, as Professor Raimondi mentioned, to have a big data. You have a big data, but if they're not convincing, they are not used. So this, I think that's a lot of things, and there are also challenges because limitations of the in vivo models. That's why we need to do a lot of in vitro and now in silico. That means computer simulation together with based on in vitro results, because that is a way we can screen out at least solutions which will not work. Mm-hmm. And then we don't need to go to this in vivo stage or preclinical, because then it's clear that it will not work.
Yes, Professor.
Yeah, I think what is also happening in the field, which is actually very interesting, and I'm just reflecting on in the last few months, is the line between device and drug is getting very blurry. And the reason is very simple. Because we as scientists want to explore more about the device, more about the mechanisms, and by doing so, we are finding that the devices we actually are using, our biomaterials, are actually therapeutic in nature. And once that word therapeutic comes into the foray, and once we have all our data on these devices, suddenly the regulators are thinking, well, this is actually a drug.
Yeah, that's a very good point, because if it is a drug, you go in completely different regulatory pathway, which costs 10 times more, and it takes 5 times more time, because to do that. For example, this morning we heard a good presentation about gallium using in orthopedic devices, which is nice. And if we saw that, okay, we have this kind which is work with bacteria to prevent that, in view on the regulator said, oh, it works like pharmacological, it's work like a drug. Okay, here we are, you go with a metallic implant, which is doped by gallium, but then it became a drug, and then you need another 10 years to get it to the market. So that's a really point, the semantic and how you formulate it, and how precisely all this precision medicine and precision scientists can work to not only to develop or to research or to study, but also to formulate at the end of the day what this particular device is and what is a primary mode of action.
And that was one of the points of Remorosa, to teach doctoral students how to think about that aspect as well.
Yes, I think that what we have in all the schools, we also have, at least what I have presented, that a lot of regulatory issues for young scientists and also for researchers to understand that this is the issue they will see at the end of their research. And it's good to know it as early as possible, so you don't develop something which, you know, after 3-5 years of research, you find out, okay, but it has no future because it cannot be approved.
Professor Alini, you already have mentioned that there are only a certain amount of drugs for diseases such as osteoarthritis, rheumatoid arthritis, and they only are effective in half of the patients. Are the bioreactors that you and Professor Obradovic are developing and testing one of the answers that can help overcome this challenge?
No. No. So, again, it's, you know, also as Professor Pandi and Professor Gatlin mentioned, you know, we use biomaterial for implant, for example. Then we realize, but we are doing something, not only the shape and replace what is missing, but we are also influencing the...
It's for the...
Just for the recording. Yeah. Okay, it is. Okay, for you. Okay. So we are again doing bottom up. So we do something, I say, but here interact, okay. Then we change that, and then and we will never get the full picture. So we miss, and the bioreactor, it's better than, say, organoid cell culture, because you use a more complex tissue. But we know, for example, that the immune system is fundamental. And in the bioreactor, presently, we don't have the immune system there. So it's an important point or player that we are missing. So after 40 years of research, I think we need to be more holistic. How? We can think, we meditate, and we go, no. I think we have some tool now that can help us. It's artificial intelligence and combinatory. All this data, I think we need to make them, as Professor Rimondini said, make sense. So until now we did one step, and then we realized that we influenced 4 steps. So, and then the regulatory say, it's not working because you didn't think about that and that. I think we have to have a different approach. So have a more complex situation, and only my feeling is only using machine learning, AI, big computer, that we can simulate hopefully, these complex situation have a more holistic approach that because, I say, the air is polluted, in 15 years you got that. Or 10% of the population, because they have a gene already that predisposes them to get that, if they get that, that's what's happened. Instead that it happened, you know, to go back to the gallium pollution, say like that, it's impossible. You know, if we don't start with some major approach. So we have to change our approach to research because, in my opinion, we are too focused on, say, one pathway. You know, can be a biological pathway, can be a bioreactor. We increase the dimension, the complexity, but you know, if we compare it to the holistic approach, it's 0.0001% still. So we are still far. So that's why I think for the young generation, I think maybe they have to take a different approach than our approach.
Please. Yes, please.
Yeah, I have to agree and disagree. So I think it will be very hard to have, I mean, I completely agree with the holistic approach, but people are so different and nature is so different. On the other hand, artificial intelligence, machine learning, and I'm sorry to really not put so much faith into this, but this is my just impression, and actually everyone knows what these IT people say, garbage in, garbage out. I mean, I also write some programs. It really depends what you intend to do. So you will have millions of data and you will interpret them, and even not you will just believe in your program that it will interpret it right. So I'm not so much, I think it has great potential, and I think definitely it will be used and give great results. But I also think that we have to solve the problems. I'm an engineer, so I'm solving one problem at a time. So if you have so many patients, cancer patients, that are not responding to chemotherapy. I knew some, it is a horrible number, like only maybe 20% of people are responding, and all the others are not responding, and they have such horrible side effects. Then you think, well, maybe there is a way that we could immediately help them. And I think if we could have a way to quickly see what is with their cancer cells, on at least which chemotherapy they would respond, that would be a help. And I also think that really technology, as everyone sees in all other areas, is really very advanced, that you have really advanced machines. I think in a bioreactor field, it could be much more developed if more people and more collaboration is between like engineers and actually understanding the language from the biology and also engineering. Because as you can see, these microchips, how high technologies are, and what people could investigate on small scale. I think if we put effort also to make some macro scale where really you could test drugs on tissue samples, or try to grow tissue samples, maybe never for implantation, but for understanding diseases or to test the drugs and to decrease the animal testing. I think this is really, it shouldn't be just discarded this way, although it is not completely holistic, but I think it has.
I conclude you both, bridging what you just said. So basically, from regulatory point of view, everything which is advanced is the worst thing you can do, because the regulatory documents and everything, they gap like 5 years, 10 years maybe, before all the bureaucracy, all the legal implications come and that. Then it goes to the national level. So every national regulator would have something to say. Now the situation is slightly different because we have health technology assessment regulation coming in force next year, which give one-stop shopping to medical device class 3 and in vitro device class D and everything like that. However, when we talk about this new, if you say something now to the Commission that you do it with artificial intelligence, you get additional ethical issues. You get additional ethical concern because it's something you cannot control. And if you have big data, but most of this data are bad data, that you get nothing but bad data at the end. So maybe we need to talk about right data and not so much big data to get it, because then at the end of the day, when you go to the regulator, they want to see the proof. And the proof, what they call now R3. This is not 3R. The 3R is for animal test reduction. R3, it means it have to be reproducible, repeatable, and relevant. All the tests you do. So whatever you do, bioreactor, mechanical, not, or drug, or whatever, it has to have relevancy for the problem. It can be reproducible, at least in 3 different labs, with at least 3 different people who can, prove that this really works. And I think that if we expand it with machine learning, that's also the case. You have to have 3 different people who do different, data feeding, and they do have to give results which can be trusted. I think that's a way. That maybe we can call it holistic.
Just a comment on artificial intelligence. That is intelligent, but not so much. And even in this case, yes, it's only artificial. We know that so the first reports of artificial intelligence was in 70 years, and so it's an old concept. The gap was that no technology was available for calculation. And in the story, in the science story, we saw some period in which artificial intelligence was up, and probably the solution for all the stuff in the world, and sometimes this was very down. Now we are on the top of the wave. The problem of artificial intelligence is that the quality of data is number one. The second, that sometimes the algorithms behind are not well known, and to trust so much on what we have the quality control may be a little bit scaring. So I think that as in all cases, we should have a clever and a critical approach and critical thinking to the data and to the results, and probably the combination of both traditional science and unbiased and then mathematical computational approach can guarantee something more than to trust only on the unbiased research.
So it won't replace the scientist, the artificial intelligence. We still need the scientist, but it could be a useful help if used correctly, right? Yes. Yes.
Yeah, I didn't say that we don't need scientists. I just said that AI can help a lot. It's maybe biased because it's true that we don't know exactly the algorithm, because it's a learning process. But scientists are also biased. So we don't have to forget that. So the bias will always be there, and we just have to really try and see what's happened. But I would say that that is a good possibility, in my opinion, to take another perspective how research will work. And precision medicine, you know, by definition is genomic, lifestyle, environment. That's why animal models for cancer drug are useless, because genetic is different, environment is different, unless we go to the cage for 100 years and then lifestyle is totally different. That's why drugs, it's 1 out of 1,000 that was mentioned, and it happened by chance, you know. There is the random, you know, in my opinion. That's, we can discuss.
And if we turn back to the Premurosa project, that's the question for all of you. You already mentioned some of the aspects and points, but what is the contribution of the Premurosa project in the development of precision medicine approach in musculoskeletal regeneration? You already pointed out some things, but maybe just if you focus on that.
The vice core.
The vice coordinator.
You are now.
Well, definitely the point is in our lab, it is this system for osteosarcoma cultivation, 3D culture with a lot of spheroids, for which we will now go into anti-cancer drug testing, so to see if these spheroids really behave similarly as in vivo, and that is our contribution.
Okay,
yeah.
thank you. Please, Professor Aki. No?
No, I think the major contribution is to, as Professor Rimondini mentioned, is to tackle certain research point that need to be developed, and to give a broad, different option to the next generation of scientists to understand or to see the complexity of the research in musculoskeletal. So I think that it was, in my opinion, we contribute to understand that we don't understand a lot of things.
That's a very good point. And I think also that what we did and what we learned from that one is that it's the things what our scientists were using Premurosa, it does not need to be very complicated, because basically like bioreactor or whatever is very simple from engineering point of view, but it surprisingly has a lot of parameters you need to work with. You need to do the precise experiment to understand how it works. And even, for example, in ALDA, what we did with the titanium implants, to develop the protocol which can be used also by other people to see how well it will withstand corrosion or biocorrosion in simulated environment, because it helps already on the stage, on the development of the implant, to understand if it has a risk in terms of being degraded too much or too fast compared to some controls. And it does not maybe seem so much exciting, like, you know, flying to Mars, or I don't know, going to Alpha Centauri, but it's anyway the practical things which all the medical device developers and manufacturers at the end of the day would ask and say how we can prove that this is something what we did makes sense and it is safe to use. So all this combination together with all this holistic approach, let's say. So it's not only holistic in terms of science, as I see, because we have here data processing, we have here biology, we have cancer research, we have bioreactors, but we also have regulatory part, and we have also engineering and algorithms. So I think that was multidisciplinarity of Premurosa was very exceptional as far for this kind of project, which normally sometimes Marie Curie project directed only in one topic, and this was very, very wide.
So from the scientific point of view, I agree with you, Mauro. The awareness that complexity is what is driven the success, I think that is very important. From the outcome related to the people that we trained, yes, multidisciplinarity is the strongest point of Premurosa. Multidisciplinarity and innovation-oriented project with the possibility even to be trained in different field and in different sector. We have, for instance, many companies inside from that help us in develop some technologies. I think that for the student it's very important because in respect with the other PhD program, Premurosa offer the possibility to see the world, as well as I would like to mention even the possibility to communicate the science that is not trivial. Last week, Nature published a nice article in which they say that the scientists are very well trained to communicate within the community of the experts, but they fail to communicate to the other people. And now we know that the ivory tower is not good for science, because the science is something that should be helpful to the people, helpful to the world. And I think that in Premurosa we try to do something to fill this gap.
So besides what has been mentioned, which I completely agree with, one other thing that we shouldn't underestimate is the power of collaboration. You know,
No, he has a microphone.
I have a microphone, yeah. So the power of collaboration across disciplines. And a consortium like that does, and Professor Boyana mentioned about garbage in, garbage out. But here we had an opportunity, all of us, to put all our garbage together and compare that garbage. You know, and then have collective garbage, which we can say, this is all our garbage, and we can validate that garbage, right? So that's one thing. The power of collaboration was phenomenal, and we should underestimate that at all, because COVID has shown us that if there wasn't collaboration across countries, across the world, we wouldn't have been able to vaccinate the amount of people we have vaccinated across the world. So the power of collaboration cannot be underestimated, and that also empowers our younger generation. Even I was a student, I did not work in any other labs. I worked in our CSIRO labs. These next generation scientists have worked in multiple labs. You know, so I love when my students go to other labs, because when they come back, they complain less. So because they know what the other labs are. They know, they have experienced different cultures, not only globally but also in the aspect of the science, and it's a fantastic training. Our best output, to be honest, is the next generation scientist. At my own lab level, I think at a very singular level, we've been able to identify a signature of human disc. As human disc is something in the back, we get back pain because of intervertebral disc. We've been able to get the first human global signature of a glycome, and working with Leah, we'll be able to identify the connection with another omic, which is the metabolomic piece of it, associated with the glycomic piece. But this won't happen if it wasn't Premurosa.
So it was a great exchange of knowledge and expertise, but also, I guess, students found friends for life and colleagues that they will collaborate later in their future careers.
Absolutely,
right?
yeah.
I hope we communicated well enough, so we have some questions from the audience. Please, now it's your turn to ask our expert. Okay. The other one, I think. Sasha Talebo. Yeah.
Don't not kill the new generation scientists with the microphone or would destroy science for the future. A comment from a more technical point, because you know I belong to the new generation. We belong, no, we are the young, very young scientists. Because I agree a lot with you when you say that we need better models, more complicated, more physiologically relevant. And I also agree with the idea that only artificial intelligence or tools like that will be able then to merge so many data, so many information. We can do it in the laboratory. But at the same time, I see that when we increase the complexity of the model, that we are unable to produce many data. Models, complicated models, can be hardly reproducible. We can give few information. So really, from a technical point of view, from the bench point of view, I see a really difficult correlation with providing relevant model and huge data. That can be, at least in my point of view, I don't know, inspiration from Premurosa II, their revenge, because I think this still a lack from, let's say, practical point of view. So I don't know if you consider this or...
So,
what was the question?
So if I understood your question, I think what is missing in the field, and I completely agree with you, is validation. You know, I think we all have models, but I think collectively as a field, we need to look at validation of not so much the healthy subject, but with the diseased subjects. So if we come up with the idea that we need to stratify patient population, we also need to think of the idea that we also need to stratify our models. And what exact question are we asking? You know, if we focus the question that not, I mean, if for example I work in disc or whatever, you know, if you work in cartilage or bone, but a model is not going to answer every question of cartilage has, right? So if you narrow the question, saying this is the stratification of the question that you're asking, and validate it with other different labs and with the human subject, which is stratified, you have a better chance of having a good model.
And I think it's also, to formulate this question, you need to know at least 70% of the answer, because you need to know exactly what you're looking for. It's not just a research question, but also which link to the patient. So you can say, okay, I can do it like this and this, and you take 10 patients, it works perfectly. When you go to 100, it doesn't work anymore. So that's a very big problem. And this problem is not only for musculoskeletal or not only for Premurosa, but one of the biggest pharmaceutical companies in the world. They have the one slide which show we are very good in collecting data, but we have no idea what to do with them. So it's a question we can easily generate a lot of things, but then to understand what we generate, what is a garbage and what is not, that's a very interesting point, because we also have to say maybe not so validate too much negative results. Negative results is equally important, maybe even more important than positive results, because that gives us a possibility where not to go.
Yes, I would like also, I completely agree actually with you, Andrea, that more complex systems and more complex experiments, less data we get, because it's harder to perform, and it's usually longer if you want it to be relevant. But I don't know, in this like race for so many data so quickly, I don't know what we are gaining, because I think if like half of the people would not work on mice, which Michael always saying how we have all the cures just for mice. If you are a mouse, you will, how you say?
The one doctor said on the conference, we can cure every disease if you are a rat.
So I think if different labs would take approaches to really be more relevant and develop these systems and to validate and to collaborate, to have similar like experiments, and then of course put more effort, but then you will have relevant data, not so maybe quickly, but they will be at least more relevant than experiments in mice.
Yeah, but why you are afraid to have quick data on human? So that's,
Nobody.
No, exactly. I think the problem, and it's the validation. I think we already saw maybe 20 years ago with finite element model, computation model, we say, we load the cartilage and we see, but we are still far from that. I hope that with, now I say AI, but it's computational, that create a lot of data, will be able to give us some indication where or how a gene that is not, that is wrong, say like that, there is a coding problem, will influence an implant reaction in a patient. So that's what we miss the link. We know that it's happened. Now, just to be simple, in the implant, that have a wear particle or not wear, or people have infection, or people not have so much main infection, or so prone to be infected. So how these, 100 genes are influences the reaction of a patient. And that's, I think, it's matter of validation, but then you have to validate it using human cases. Because we know, we have a lot of data. We don't know how they are related, but we know that this patient has infection. Okay, do a sequence of his gene, take his lifestyle, take its environment, if he's living, in a healthy, what we consider healthy, in a green forest or whatever, maybe it's worse than, but, or in a polluted city. Is that a relation on that? I think with this big data analysis, we'll be able not to solve everything, and then we have also to give time to this new technology to develop and to get even more, I would say, precise, not powerful, but precise, and give us.
It has to be powerful.
Yeah, as a fast interaction and bite or beat or whatever. So I think validation it's fundamental. It will not be easy, but we have to change our approach. I think we are in a same, as a 20, 30 years ago, get vegetarian or not. It's a life decision. I'm not, for example. So I just, or drink and not drink. You want to have, it's a completely approach of your life. And now we are in a similar, in my opinion, situation where if we don't make our change in our research approach, we will be stuck there for the rest of your 40-year research, and So that's my opinion, and maybe I'm wrong, and probably I will not live long enough to see if I'm wrong or right. But that's my suggestion based on my experience.
Thank you. Do we have some other questions? Yes, please.
Hi. So we heard a lot of the good thing about the new generation of scientists is that they collaborate a lot and they bring together a lot of these fields. And I was wondering, if we are looking at how all of these technologies are being regulated, do you believe that there needs to be some sort of evolution in that field as well? Or how do us young scientists navigate this potential minefield in order to get our new technologies or approaches to market, basically?
Yeah, I think that is what we have discussed and what we try partially to do in Premurosa, to talk about the regulatory science, to understand, to give at least you an overview, the first introduction, just to feel what it is all about, because the regulations is something which is usually outside of our control. It will be done by lawyers, by specialists, by experts, by some committees, which we have very little influence. And then when it go in the law, then you cannot do change too much. So then it's done. And I think that one of the approach which is now being more diverse, at least in European Union, is that the more precise formulation we have, because before we have drugs, we have medical devices, and we have the what is called ATMP, Advanced Therapy Medicinal Product, which is cell therapy and gene therapy, everything like that. And there has been a lot of products which does not fit in either of that. And if you have like drug device combination, that's a headache, because then you need to certify two of them, a device and drug, and to prove that. So you have a double work, and that was the reason why nobody was too much interested from manufacturer point of view. Now we have SOHO regulation, species for human origin. Now we have health technology assessment regulation, which takes 9 paragraphs or 9 dimensions of all medical devices. So it's not only performance, clinical performance, is safety, it also says social aspects, ethical aspects, economic aspects. Does it decrease the burden of the healthcare? Does it improve the quality added life years? Does it have a good incremental cost effectiveness ratio? So we need, I think, besides the regulatory stuff, to more to social scientists who involved in the ethics, in social system, in healthcare system, because we don't know how it works. Okay, we use it personally. When you go to doctor, you interact with the system, but what it is organized, how it's organized, we have no idea. And it may be not our function to know, but we need experts to talk about that. So maybe in the next project we can have more inclusion of these people who help us, and also people from communication, of course, because all this communication semantics needs to be very, very clear.
Yeah, I think what I would add to that is, I wouldn't limit your innovation only to devices and drugs. You have tools that you have learned in the training process. It doesn't mean that limitation is device or drugs. You see in our field, for example, people are growing meat in the lab, and there are companies now which are manufacturing meat and selling them as vegetarian, vegan-based, mass-produced, meat-based instead of going to animals, right? So that's the innovation that has happened from our field, by the way. It's not about a device and implant, right? There's the whole field of organoids is bursting, huge. Roche has invested billions in that. We all know
and
that.
What is organoid? Is it the drug or is it the
Yeah,
device?
But
or
it is.
what?
But at the end of the day, it's used for testing substrate. It's not, But so the innovation, don't limit your innovation to saying it has to be an implant. I'm working on regeneration. No, but there are hundreds of other things one can think of. I'm middle-aged, so I don't have much ideas anymore. But you guys have, there's so much opportunity for innovation, incredible. And with the use of AI, it opens a minefield.
I think for you as a that you finish your PhD, I always look at my PhD as a school. Learn how to solve a problem.
Yeah.
Where at my time, where to get the literature, where to get the information, and how to approach. But that doesn't mean that you have to stick to your field. In my opinion, more you change, better it is. Because you change your approach, you see other things, and you can bring to the new field what you learned before. Because if you stay 40 years in your glycan, you will get sick after, to see this network in the shoe. So, but you can use your knowledge to develop another field of research, and that's, I think, it's where the innovation will also come. By changing, by see other things. If not, you stay in your black box.
Get out of the box.
We have time for a last question. Yeah.
There. Two questions. Two questions.
Okay, 2 questions. Mine maybe is fast. So 20 years ago, let's say, you were great if you were a great scientist. But today, all of you are scientists, regulators, administrators, human resource management, lawyers, great in semantics, and all that can fall in the water if you don't know how to get the money to get the new generation of scientists. How hard was it for you to pick the 13 new generation scientists for Premurosa and for any other project that you were maybe in future going to work on?
It's a Leah question.
We have Excel file.
I like to learn something from different fields. So I feel not a burden to have to manage even different stuffs. I think that you, as a young scientist, have to know more or less what is the needs, and you have to ask to expert to improve the level of knowledge and the contribution inside the project and so on. For my experience now, just recently, I participate to several consortiums and apply to big consortium, big grant to European offer by European Commission, and the problem was the integration of social sciences inside the scientific part. So I think that we just change the way we should do science to do something useful. And something useful is to take care even of how the society is organized. And so I think that if you want to be a leader, you know the world and how it works. No
You need to be able at least to fix your computer without calling IT support. That's very, very important.
I just wanted to mention something regarding the regulatory point of this regulatory science, because, okay, before I was working at an agency in Slovenia, but was working for EU Innovation Network. This is the new just group that is, for the moment, they are working on this, try to build together academia, clinics, and regulatory. And regulatory part would like to help at earlier stage of the private company, which obstacles are needed just for that you can finish to come to the practical, to the market. So this is the new group, so that is nice to contact the agency.
Which group you mean?
EU Innovation Network.
Yeah.
And that was also inside the pilot study, just like advice and providing the advice for specific parts of the project, for example, or these different, especially for tissue engineering, and because we understood, of course, that science is big. It's not only compounds, it's also medical devices. It's very complex. To bring together all different people that are experts, and it's important to collaborate more together. And we had also big event at the end of October 2023 in Dublin, that where we came all together. That was the part of organization too, and it is online on EMA website. So you can listen to these presentations and all the questions we're sharing. So it's good.
There are basically many groups which can help you. If you have a European project who won't commercialized, there is Horizon Result Booster, which helps to do that. Then if you just have a drug or medical device whatsoever, you just call ITF. Innovation Task Force or EMA, it's free, and they can arrange an experts which grill you in the pitch session and say that way, and give you very clear words to understand that what you're doing is not useful or something. But on the other hand, you can get, especially with new regulation, you can get joint scientific consultation, you can get joint clinical assessment for the device and everything like that. So there is a lot of different ways how you can do that.
Yeah,
I'm
for
sorry
the
to interrupt
had
you.
every moment.
No, sorry, I'm interrupting because we are running out of time, but there's going to be time at lunch to continue this discussion. Thank you so much to our
Thank you.
experts. I hope there's going to be a Premurosa too. Yeah? No? Maybe?
No, we
Two,
Why not?
yes? Maybe there's an idea.
That's a big secret.
Thank you to our audience here and online, and there's going to be a chance, of course,
the coordinator doesn't know.
continue this debate. So thank you so much.
Thank you. Thank you.
Thank you.
Thank you, everyone. Thank you, everyone. Thank you, Petra, for leading this beautiful round table, and for all the guests. Hello, welcome back everybody. I hope you had a nice lunch, coffee, break, you're relaxed and ready for the next lecture. So, now with us is Enrica Verne. She will introduce to us what are the bioglasses or bioglasses.
So the pointer is this one. Yes, okay. So thank you for the kind introduction. Yes, the title is quite strange. Probably you are asking why I changed the font in the word bioglasses. For a very simple reason, because bioglasses belongs to a special class of materials that can be used for bone substitutions. In the area of materials for bone substitutions, we recognize 3 different generations of materials. At the beginning, with the initial definition of biomaterial, people working on this topic just look for bioinert materials, looking for substitution purposes, just to substitute some portion of bone. Then, by the end of the last century, the importance of the biological impact of this material with the human body reached much more importance. So, who studied these materials usually took very focus their attention both on the material questions like mechanical properties, composition, processing, and so on, but also on the impact that these materials have on the living organism, but with a more precise purpose, which is integration. So this is the era where bioactive materials have been invented. So materials that are able to interact with the body, modifying their surface, and promote the integration of a graft into the bone. After that, from the beginning of this century on, the research focused much more on the bio problems, on the bio aspect of the interaction between materials and bone. And we can talk definitely about the third generation materials that are often smart biomaterials that have regeneration purposes, not only regeneration, as we will see in brief. So why glasses? Glasses for bone surgery may be that people who is in this room already know it, but for general people, general public, could be a strange thing, how a glass can be used to repair or to integrate or substitute a bone. So these glasses exist. They are called bioactive glasses or bioglasses, and they were discovered in 1979, more or less, by Professor Larry Hench, who works in Florida, and he invented the first bioactive glass composition that is very similar to the common glasses that we use every day, so silica-based glass, like windows, glasses, bottles, and so on, but with a special proportion that can induce a bioactivity mechanism when put in contact with bone or when put in contact with the simulated body fluid that, in few words, induce the surface modification here with gradual transformation of the surface of this material into something that is enriched in calcium and phosphorus, and then at the end crystallize in form of hydroxyapatite, which is the mineral part of the bone. So if we see, if we observe the surface of these glasses at different time of immersion into a simulated body fluid, for example, we can recognize the different phases and steps of bioactivity. So what happens after the discovery? Very quickly, these glasses go to the market. So it was open the clinical application area where the goal was bone tissue integration. So people working on this topic make a lot of efforts to optimize the composition in order to optimize also the bioactivity mechanism to induce bone integration. And several commercial products reach the market. They were always particles or small bones, like the middle ear bone chain, or granules or little bone substitute. From the new century, the beginning of new century on, even bioactive glasses have been studied, taking into great account the biological impact. So we didn't invent anything very, very new about the composition to be bioactive, but we work hardly to optimize the composition in order to have not only bioactivity ability, so to integrate with bone, but also regenerative ability and therapeutic ability. And we can do it, people who work in this field can do it by doping the glasses with a variety of ions that it is proven that can be useful not only for bone generation, but also for example, angiogenesis, anti-inflammatory properties, antibacterial properties, as we learned before. So my research group started working on bioactive glasses in 1992 when I graduated. And we started during the evolution from bioglasses, when also the composition was important, to bioglasses where the biological impact was the most important thing. Doing what? Okay, at the very beginning, we try to find alternative composition to the first bioactive glass invented by Hench. We normally use melt and quenching route. We can prepare some bars of glass that can be powdered and then used to prepare something like, for example, a coating on a dental screw, or coatings on inert ceramics in order to make their surface bioactive. The two is here. I don't know why. And then we move to the preparation of bioactive glass scaffolds or glass ceramic scaffolds, and we started with a simple way that people around the world also begin to use. So we explore the processing that used a polymeric sponge, like a sacrificial template. If you look to a polymeric sponge like this at the scanning electron microscopy, this is, for example, a polyurethane 1, like that, you see that this morphology is very close to spongeous bone. So what we did, we take this sponge, this one, the yellow one, and we put the sponge into a slurry containing a liquid phase and glass powders. Then, when the sponge is drunk, we have another morphology. So the glass particles are all deposited on the struts of the polymers. And after a thermal treatment, we obtain the ceramic or glass ceramic replica of the original sponge. With good mechanical properties, we can drill, and also able to keep by capillarity a lot of very quickly liquids, because the porosity is completely open. If we, che succede? Che non va avanti. Okay, if we, enlarge this is a strut of this, and this is what we obtain when we put the scaffolds into simulated body fluid, so we see the growth of hydroxyapatite. And if we seed bone cells, bone progenitor cells, or in this case probably they are osteoblasts, we can see that not only the cells are in good health, they survive, but they also start working, because these different morphology, like this, not little one, but the big one, globes, I need a new bone, are produced by the cells. So they are starting to produce new bone in vitro. Next generation of scaffold processing pass through different ways, like foaming of particles. In this case, glass is obtained by wet chemistry. We also use other sacrificial templates, like, for example, stale bread, or other approaches used to cover the surface of biocarbon scaffolds obtained by plants, by wood. And of course, now, from in the last years, we move also, as people working on this topic, not only we, of course, on additive manufacturing technologies. In this way, it's very easy to tailor the scaffold properties. We have a very good control of the process parameters. This is, for example, an example of robocasted glass ceramic scaffolds that we prepare and characterize in collaboration with Tampere University and the Innovation Center in Iceland. But glasses are not only bioactive, they can be also therapeutics, so they are bio-bold glasses. We talk today about infections, and it is very easy to dope a glass with something that go against bacteria colonization. We did it on our glasses by ion exchange, so by putting the glass into a solution containing soluble salts of silver. Silver is well known to be an antibacterial ion. So what happens? It happens that bioactive glasses can leach ions, for example, sodium ions, and that are exchanged with silver ions. In this way, you can enrich only the surface with an antibacterial element or ion in this case. And what we can do is to tailor different parameters, such the time, the concentration of the solution, and the temperature that we use, in order to tailor the concentration and the deepness of the silver-enriched layer. So, in this way, bacteria are not so healthy. This is a very, in our opinion, interesting technique, because we can apply it to bulk materials, but also on as the last processing step on coatings, for example, for metallic prostheses that we want to make bioactive and antibacterial, on sintered samples, so bulk with better mechanical properties than the glass one, or scaffolds. These are three images that let us see that bacteria don't grow around the material charged with silver. We can change and modify the surface of a glass, of a bioactive glass, also by functionalization. And we explore a lot of different kinds of functionalization. Very recently, we focus on the functionalization with biomolecules, like tannic acid, gallic acid, or generally speaking, the polyphenol family extracted from vegetables, like green tea, grapes, and other vegetables. So we extract these polyphenols that are in some cases antibacterial, antioxidant, anti-cancer. They have a lot of interesting properties, and we graft these biomolecules on the bioactive glass. We also found that not only these molecules have these interesting properties, but they are antioxidant. So antioxidant means able to reduce something to something else. For example, they can reduce silver ions into a solution to metallic silver nanoparticles. So we can choose if we want to use the ions or the metallic nanoparticles. The topic of doping bioactive glasses with the therapeutic elements is now greatly developed, and we use a lot of different ions to dope different glasses with different morphology. So now we are able to prepare glasses with anti-inflammatory properties, antibacterial activities, osteogenesis activities, and angiogenesis activities. Some of the composition that you see into this table has been object of the studies of our PhD, Mari, in the frame of the Premurosa project. But knowing how to dope a glass and also how to functionalize it, we moved from hard tissue, like bone, to soft tissue engineering. Because if we prepare glass powders, for example, doped with therapeutic ions, and we will see later on also functionalizing them, we can also use the glass as particles, nanoparticles, to be dispersed into polymeric matrix like hydrogels and so on, to prepare membranes that can be very useful in the treatment of skin, for example, for skin regeneration. What will happen in the future? We are still working on this topic. My colleague Francesco Baino is the PI, local PI of a couple of projects, one national project and one Marie Curie project, where the scientific goals is to improve the specificity of the glass ceramic or glass scaffolds using different techniques, for example, stereolithographic methods, together with in silico models developed coming from files obtained by images of sponges or bone. So you can also prepare by 3D printing or a trabecular morphology. And my colleague Marta Miola also now is in charge for a national project that is concern plasma treatment of the surfaces or of ceramics and glasses, and with this technique we try to improve the surface properties of glasses, even if they are powders, in order to reduce bacterial adhesion. And to conclude, we recently have been involved in a national project, very large, where our little contribution will be about the realization of bioactive nanoparticles or microparticles that we will dope with therapeutic ions, and or we will functionalize with the tannins and natural polyphenols in order to prepare powders to be introduced into a polymeric matrix and to be and to put this construct in contact with a biological model of diabetic foot. So we will try to find a new approach to favor diabetic wound healing. So if you have this question in your mind, this is the answer. We can reconstruct a bone or a tissue with a glass. Thank you.
Are there any questions?
Okay, thank you. When you show that this glass, which is also doing for skin, for soft tissues,
yes,
this composition version, do you think you can do tendon?
So someone investigate the possibility of creating fibers, glass fibers, in order to guide other kind of cells like neural cells, but they are not thinking about tendons, but
because in tendon, I think one of the option problem is to get the osteotendon connection.
Yeah.
Because you have a transition here from hydroxylapatite to more soft tissue, like that's one. So maybe you can, if you're interested, you can join TENET cost project, because it's about tendon regeneration, just started a year ago.
Thank you for the information.
Yeah.
Yes, I'm
Good.
very interested.
Thank you.
Thank you. I would like to ask you more in detail about the approach to the diabetic foot wound healing. What is
okay? We have to start already, so we don't have results, very significant results at the moment. But the idea is trained by the topic of the whole project to prepare digital and biological twins as models for some kind of injuries. One of them is diabetes, and we work not on the injury but on the comorbidities of these injuries, like the wound healing of the part of the skin that is damaged for this illness. And to do that, we are in contact with a couple of colleagues that are growing a skin model. And when we will be ready with our membranes, we will try to put the membranes on these skin models and see if they are able to participate to the metabolism of skin and skin healing. This is more or less the idea. You're welcome.
Very nice to see your journey to date. I don't know much about bioglass, but have you looked at the crystalline structure of bioglass, or the context is there any bioelectric or piezoelectric properties? Have you investigated that, or?
We would like. I really talked about that with my colleague Marta in the last month. We would like to prepare piezoelectric glass ceramics or something like a composite with glasses and something like piezoelectric, because we know
the
that
reason why I say is because
bone is piezoelectric. So
yes, I know. Yeah, that's why I asked the question.
So yes,
yeah.
It is a challenge topic because we have also to face to biocompatibility. So
it could be a good substitute for carbon nanotubes, I think, in some ways, if you can do it that way.
Or if someone can do for us.
Yeah.
Yeah.
Nano PLLA are also piezoelectric,
yeah.
so just coupling the two
We can
materials
try.
maybe.
So I know more or less than a beyond bioglasses, but are the biocompatibility improved since the beginning? You know, the silicium release, and the strength are still a problem, or is still a problem, or now with the different composition has been improved? And they are mainly on dental or also on bone?
I start with the last question. They are intended for bone substitutions almost everywhere, almost. Of course, we need mechanical properties, but the new generation of scaffolds present mechanical properties that are very close to bone, new bone, healthy bone. In fact, we can drill, we can fix with screw to the bone, also micro screw for dentistry and so on. And the biocompatibility, it is a question from the beginning of the history of bioactive glasses. The problem, in brackets, is that when bioactive glasses start to exchange ions and keep ions from the solution, the pH changes. But this is mainly in static condition. In dynamic condition is not so, how can I say, crucial. But if we also modulate the composition, we also can limit the pH increase. For example, glasses containing also much more phosphates are not so impacting the pH around, because they also can buffer the pH by leaching phosphates and together with the other ions. So the problem of biocompatibility, I think, is not so crucial as once upon a time. But I think that it will be useful to make a round-robin test with different laboratories to, how can I say, to suggest a standard procedure to test the biocompatibility of such materials that dynamically change their surface and the environment around. So it is very difficult to find, for example, a control, because what can we use as control? An inert material? I don't know. I think not. Probably a bioactive materials which is already on the market. We have to decide that, because literature about bioactive glasses is very rich of biocompatibility tests, but not all are comparable each other.
But 3-calcium phosphate or TCP or other biomaterial that is used, granule or like that, are still better, so in the sense that are more used than bioglasses. They probably are not active.
It depends on what we intend by active, but they are, sorry, conclude.
No, so I'm saying are still mainly TCP or all these hydroxyapatite like biomaterial that are used more than bioglasses, or there is a market also for bioglasses?
Yes, there is a market also for bioactive glasses. Of course, the other phosphates are well known, more than bioactive glasses, not for a scientific point of view, but for the market, of course. And, but glasses are not better or worse, are different because they are amorphous. You can dope them in a different ways. You can adjust the composition without affecting so much their bioactivity. So they are very easy to process, to design and process. So someone likes glasses and some other like ceramics.
Thank you for a nice talk and discussion. And we are coming to our last speaker, Abhay Pandit from the National University of Ireland. He will introduce us to the topic of personhood, understanding the identity of tissues for therapy.
So it's a very different talk that I normally give. So it's I was asked to give a TED kind of a talk, so that's. It's there, Slovenia AP.
Oh, no. Is there a presentation? Not.
Second
okay,
last,
yeah.
yes.
Sorry.
So great. I'm going to talk about personhood, and I'm going to start me as a person and what has influenced me, what I do, and the influence that brings me up, and then bring that to the context of, if you take me as an example of my identity, how even our cells have their own identity. So that's the bigger context of this work. And I'm from Curam, the Center for Research in Medical Devices. Curam actually means care in the Irish word. So just so you're aware of it. I like murals. I love murals. The reason why I like murals is because it is accessible art. It's accessible art where you don't have to go to the museum. Everybody can watch it. It's not about, there's no elitism in artwork. So throughout this presentation, you'll see a lot of murals. This is a famous one by Bansky, and I was glad to see in Slovenia I was delighted to see a poster of Bansky's work that's being shown. So anyway, so this is Bansky's painting, which was donated to Southampton Hospital, saying, thanks for all you're doing. I hope this brightens the place up a bit, even if it's only black and white. And also used to this presentation in the context of post-COVID. And the reason why COVID is important for us, because that's when Prima Rosa started. I remember going to Italy in January, I think January or February, and then March there was a COVID started. So there is a context in which I placed this whole presentation of, and this is where Bansky is thanking everyone, saying that. I also want to acknowledge Prima Rosa to take care with care. Kuram is care. So to take care with care, no wonder I'm a part of this. And Maciek Dozik, who is a graphic artist who helped me with my graphics, and Ed, who is one of our ESR as well. I also want to thank all the taxpayers funding it. That's why I'm standing here, because people pay for what I do. Research-wise, industry also pays, and also patients who tell their stories to make my work more meaningful and donate a part of their personhood so that we can understand what those tissues are. So I want to thank them, all of them upfront. I want to have some caveat. This is a street art in Ljubljana. As I see some nods in the head. All this presentation is all about personal opinions. I do have a bias. I do work with industry. A lot of data may not be my own data. It's not primary data, and I have used analysis from peer-reviewed sources. That's the caveats behind this presentation. So this is where I was raised. Thane is in India. It's very close to Bombay. When I grew up, it was called Bombay. Now it's called Mumbai and I wouldn't go to the reason why it's called Mumbai, but nevertheless, I still call it Bombay. Bombay is a Portuguese word. Bom is good. Bay is good bay. That's where it is. And the first railway line, India has the highest network of railways in the whole world, global world. That's because the British built it so that they could transport everything to make their life easier. That's what they did. The first railway line was 32 kilometers long. So Thane is where I'm from, is 32 kilometers long. Now if you go from Bombay to Thane, you won't know the difference. So for all practical purposes, I was raised in Bombay. This is the city I was born in. It's a city of lakes, lots of lakes. This is the theater. It's a performance venue, and my apartment rather, is right here, which is where I was raised. And my current apartment, which I bought for my parents, is in this tall building. I'm not showing off, and I'm saying this is just a fact that where I am. This is the micro environment I was raised in, and these are my parents. Unfortunately, they're no longer alive, but they're there. So who am I? I did not get into medicine. I wanted to be a doctor, medic. I did not get into it. I got into engineering, biomedical engineering. It was the first program in biomedical engineering. I didn't like engineering when I got into it because I wanted to go in medicine. They didn't take me, so I happened to be in biomedical engineering. So I barely passed my first 3 years in engineering, and in the final year I came out with the first class. I don't know why. I think there was a mistake, but nevertheless, I did come fourth in the whole university at that point. But everybody wondered what's wrong with him. Why did he do it? I joined a master's and PhD. I actually failed in my first years of my master's because I didn't really like the field of biomechanics. Excuse biomechanics people. I didn't like biomechanics, but nevertheless, I switched universities and then I did a PhD, ultimately in the field I love, which is biomaterials. I did not want to be an academic. I did not want to be an academic at all. I hated to write. I don't want to be like these individuals who are narcissistic, who write grants, who write all over their papers. I didn't want to be any one of them. So I went to industry first. I chose to go to the industry. I said I'm going to work for a big company, and I worked in, and actually I didn't even apply for jobs. They came to me. I was presenting in conferences, so they came to me saying, okay, so here's will you work for us? So I worked for 3 companies. The first one I left. The second one was a startup, spin-outs. Spin-outs go under after 2 years, so I was laid off. I was told this was in the US. I was laid off, so and they said goodbye. It was a startup tissue adhesive company. But nevertheless, I got another job in a heart valve company, again, a very blue sky project on tissue engineering heart valve. No wonder we don't have any heart valves anywhere in the market, but it was a very high risk project, and I was laid off from that job. And by then, in the last job, I had decided that my place is actually in academia because I like these blue sky projects. so I better go back to go to academia, because that's what I actually like to do. And I don't want to be like what I don't want to be, which is X, Y, and Z. I may very well be that now, but I don't want to be who I am. So I did get a job in academia without much effort, because like Ilyana, I also had 9 papers from my PhD. So it just happened that I was quite productive in my academic. So my academic job was at that time was quite easy to get, and I ended up now as a professor in biomimetics. So I've lived in India for first 21 years of my life, in the US for 14 years of my life, and for 22 years in Ireland. So this is what my identity is shaped by. So this is who I am in my own microenvironment. The point what I'm trying to say is every one of you is also shaped by that experience. It's not good or bad, it's what we are, right? So it's not, so if you look at me, you may think I'm from India, which I am, but I've only spent 21 years of my life in India. I've spent 37 years outside India. So who I am doesn't matter, but so everybody has that identity that we refer to, and that's important to know. From an individual level, and cells and tissues also have that specific identity that we're going to talk about. So I'll come back to that later on, but more importantly is just want to talk about medical devices first. If you look at medical devices, they were all fairly accidental. It wasn't like we are going to research science and we'll come up with medical devices. They have all accidental in nature. The best example I can give is intraocular lens. This is really, he was an ophthalmologist in the UK, working in the Royal Air Force, and there was and he had a patient who came in, and previously they were using glass, not bioglass, but regular glass as intraocular lens, and they used to get cracked and everything else. But there was an accident of a Air Force plane, and then there were a lot of shards of PMMA, polymethyl methacrylate, which were embedded in the patient's eye, and he found that they weren't having any reactions to that. So there was no foreign body reaction at that time to that piece of glass. And he said, well, now I'm going to investigate that. So it was accidental that Ridley came up with the idea of, let us make intraocular lens made up of PMMA. It wasn't that I'm going to investigate PMA. It was just that this accident happened, and then it is one of the highest selling implant in the world right now. We also have to remember the era of after the World War. This whole idea of mass manufacturing. If something works, let's manufacture everything mass. So whether it is fabrics, I think I'll start with textiles, because medical devices are mainly textiles in nature. Obviously there are metals, but mainly textiles, and most of the people in textiles were women. Actually, interesting to see that at the ground level. Most of the bosses were obviously men. Maybe even now that is the case, but nevertheless, the textiles that were used were PTFE and Dacrons. All of those materials were mass-produced materials. How do you find the use to that came through that? So there, so obviously it started with fashion. Women were trying different kind of materials, but so were the men as well at the same time in the 1950s. And and colors came into being. We had these dull colors in the 1940s, but in 1950s all these colors came into the fashion. But that color is only possible because you had synthetic materials, and those synthetic materials are also used in medical devices per se. One size fits all was the mantra. One size fits all. Let's make everything the same size. It will fit all, and we know it doesn't. We know that things that are meant for men doesn't necessarily fit well for women, or it doesn't necessarily fit for all. So it's one size fit all was the mantra then, and that started with insane. I mean, good example again is the dialyzer, which was made designed by William Koch, was a washing machine. Kind of, you just take, you take the eluents out, it's a washing machine, you clean the blood out. So you take the blood, clean the blood, it's like a washing machine kind of idea. But again, it's one size fits all concept was predominant. So it's mass manufacturing, one size fits all. Remember those concepts all came from the Industrial Revolution, all those ideas. So current medical devices, there was no rocket science in that piece. Materials, mass-produced materials, one size fits all. No wonder a lot of these implants are failing in the market. It's not surprising, And the reason why we know they're failing is because we are living longer. Just that's another piece you need to think about. I did some little bit of research on Slovenia. I also realized Slovenia is one of the global leaders in medical laser systems. Medical devices are also important. So we are in the country where medical devices are actually quite an important, significant part of the Slovenian economy. And it's important for us to know that when we're here, we also recognize that there are opportunities in biopharma, gene and cell therapy, and other measurement devices. So I just wanted to bring that to the context of where we are speaking and to the audience we are speaking. Slovenia is a fairly significant player in the medical device space. I can send you the slide later on if you want. We are an aging society, whether I like it or not. I'm going to age. I'm going to live longer than my parents. My parents lived longer than my grandparents did. In 2050, 1 person in 3 will be over 65, and 1 person in 10 will be over 80. That's significant. That is very significant number than today. So we are aging. We are definitely going to be an aging society. It's not a negative thing, but that is a fact. What is also a fact is that we are going to have chronic diseases. We are going to have chronic conditions inherently present. My parents had, my mother had diabetes. My father had high blood pressure. So those genes are inherently there in me. So I may very well get that as well. So it's not whether I choose it or not. It's there in my identity. It's there in your identity as well. It's there in your tissue architecture. One good example I have is about insulin. Insulin was invented in 1920s as something that prevents death due to diabetes, right? So people could live with diabetes for a long time. But what does happen with diabetes, it's not only about living, but there are long-term effects such as blindness and others which are decoupled from insulin. Right? So one could have diabetic nephropathy, diabetic blindness associated with diabetic, even if you can live longer with insulin. So we could live long, but we could still have other side effect or chronic issues associated with that. So that is something that we need to bear in mind. The context of that is the medical device that we put in, the demands are higher. The hip you had put in when you were 50s, it has to perform till you're 90 now. Previously, people age limit was 20s, 70s, so it was fine, but that hip has to perform till 80 or 90. The heart valve that I get, I don't have one, but if I do get one when I was 20s or when I was 15, now has to live till 90. So that's a stretch. So just remember that context of medical devices. However, there is hope. So Edward Teller, who's a Nobel Prize winner, who invented the hydrogen bomb, he actually got a Nobel Prize for peace because he advocated the peaceful use of hydrogen bomb. Edward Teller says today's science is tomorrow's technology. So what you heard today, could be the technology for tomorrow. So when we talked about innovation, that's where it comes from. And I like this. And again, this comes from the COVID space in science we trust. So we have to trust our science to do that, and there is hope in that science. So the complexity of individuals also is embodied within our body. So you have our tissues. Our tissues are made up of cells. Our organs are made up of tissues. All of these are very complex entities. Just like my own environment has been complex, your environment is complex. The environment of these tissues and cells is also very unique. It's not one size fits all. It is a very complex entity, and we have to recognize that in the field, that this complexity is something that we need to understand from day one. If you look at the cell membrane, this is a cell membrane, or a cell membrane. Again, the complexity of all of these components is huge, and these cell membrane changes from one cell to another cell. It's not the same. So the complexity becomes much more larger at that molecular level, and understanding that is also key to what we want to do. So what we want to move here is, I brought you the concept of from mass manufacturing, we want to go to the personalized medical therapy. So we want to identify, more importantly, the right device and the right fit for the right patient. I should have placed it the other way, is you need to find the right device for the right fit and for the right patient. It's not for all patients, and more importantly, at the right time. So it's important to know the four rights: right patient, right device, right fit, right time. So let's give you some examples of where the research and where we are influenced by. We as scientists, we are often influenced by the stages of salamander regeneration. Most of you know that if you were to dissect out a salamander limb, it will regrow. But mind you, these salamanders are healthy. Just a caveat there. These are healthy beings, and if this dissected all, it's a healthy wound, but this naturally regrows. So there are cues in the healing process that we are inspired by, and tissue engineering or regenerative medicine facilitated by biomaterials also takes that understanding of how regeneration occurs. And this is one example thereof that we can look at. We also look at cell identity. We know that cells, whether it's stem cells or other kind of cells, have a property to repair. They may have a property to replace. I'm not 100% sure about that. They could have a potential for regeneration, but they definitely help in the repair process. More importantly, cells secrete stuff. We call that the secretome. And they may have endogenous sensors. They may have sensors that will sense the microenvironment. So this secretome is therapeutic in nature. We know that from healthy cells and from stem cells, for example, you can have a very good secretome which could be used in a therapeutic sport. So cell identity is important. If you have a particular cell identity, will give you that right level of secretome. So again, it's the identity issue in the question of cells. So we can, as I'm a material scientist, so we can define the cell identity through many different ways. One is providing structural cues. We can put cells on different structures and we can dictate their cell behavior, or we can do with compositional cues such as taking a material and changing the cues thereof. So in Primorosa, for example, we have taken personalization was important of tissue structure. So we have looked at how can we create by looking at acoustic stimulation of cells, how can we provide that tissue structures? Can we predict computationally the structure that we can get, or can we look at mechanical loading to get that tissue specific structure, or maybe precision cellular guidance can be done by different methods such as substrates and X, Y, and Z, or composition nature can be, and this is what we have been working in Primorosa in that context, but mainly taking a personalized approach towards a cell identity space. And so we are going now in our field at a phenotypic level, right from understanding of DNA, RNA, molecular, cellular, tissue to the organ scale. So this is the scale we are looking at, and fortunately we have fantastic tools to work off, and this is what we in the field are looking and probing thereof for the first time in the biomaterials field, is looking at what is the protein that's associated, what is the metabolome associated with, what is the RNA, DNA structure, and what is the genomics associated with that, and we are trying to link all of these omics, if I were to call the word omics, into that, and we have fantastic tools today. So the field is rife with the scale that we want to go to organ, to the DNA level, make that connectivity with the informatics thereof and the tools thereof. So from an AI perspective, there's ample there for you to look at. I mean, there's plenty of tools there if you have the right data. So I'm going to start my own personal journey in that space. So I started my work in late 80s with looking at what is the host responses to medical devices, and then went on to look at how can we improve these medical devices by pharmacokinetics way in terms of drug delivery, and then looking at what the mechanisms associated with the medical device. So this has been my journey, is all about host response, and that's what I've been doing almost all my life. But what I've realized over the years is that there is something that we've been missing in the picture, and the picture for me at least was that cells are covered with this whole huge structure which we call sugars, and we haven't really probed the structures well enough. So I've been fascinated with sugars, and I thought maybe there's one thing if I do in life right, I'll just do at least this piece right, because there's so much else that can be done. But let me just focus on the sugar structures in this case. So these structures are present. This is a endothelial this is the capillary, and around that also you see this glycocalyx, we call it. Glyco is sugar, calyx is what surrounds it. So the sugar has not been researched in the field. So that's what we've been doing over the years, is to understand this glycocalyx. And the glycocalyx is not as simple. It's very complex, and I'm not going to go into details of it, but these are highly branched chemical structures, and they're very highly transient structures. So no wonder they have not been researched, but this is what we've been doing in understanding the disease context. And for a long time, there weren't any analytical modalities to understand this sugar structure, and that's what we've been doing. Is now that the tools are there, can we probe these sugar structures, is the question. And the difference in sugars and proteins are driven by DNA, RNA. So in other words, there is a template that drives it. Sugars is not. Sugars is not proteins driven. It is a result of some metabolic and enzymatic reactions that happen at a cellular level. So that's the complexity that we have to grapple with. So in the first attempt, so this is Eric Shepard's work. So Eric looked at healthy intervertebral disc. Intervertebral disc, for those who don't know, is between the two vertebrae, and when these discs get degenerated, they hit on the spinal cord, and that's where people scream, "I'm in pain." So the pain is what triggers the patient to go to the clinic and say, "I'm in pain, what can I do?" And most of the underlying cause is the intervertebral disc. Discs are made up of sugars, mainly predominantly sugars. So we thought this would be a great target to look at what is the sugar change with disease. So what we did, we got donations from human subjects, healthy and degenerated, and we looked at different equal gender population. And what we did find that across healthy and disease, we do see differential expression of sugars. In other words, the sugars had changed its structures from healthy to disease stage. And not only one, but there's several sugars that had changed the structure. I'm not going to talk about which kinds of sugars in the interest of time, but this is just a summary of what he's been doing over the years. And this is the first ever study in the world to talk about that level of changes that we see in human subject versus healthy versus disease. We've also done a similar study with Parkinson's in the same way, and this is just to add that in Parkinson's as well, that we find different sugars, not what we saw in discs. So again, the differentiation between tissues, it's not that these sugars are differently expressed, means in Parkinson's the same thing happened. No, there are different kinds of sugars that are expressed, and these again the human sample study with 30 patients, and this is in Parkinson's. We did see differential expression of sugars in different areas of the brain in Parkinsonian patients. So again, different things. So what do we want to do with the sugar? So we want to get this signature. I call it a signature of the disease host. And I want to come back to material science some point in my life and saying, can we create an implant or a material which will engage this diseased host, not the healthy host, disease host. And we know the signatures of the disease host now in different stages. In discs we know in Parkinson, and we want to create this engineered material which will engage with the disease host. And this is what I mean by personalizing for the disease. This is right now a hypothetical example of it, but this is the real example of the disease host that we know. So this is just one step at a time for us. So it's all blue sky right now, right? It's total blue sky work that I talked about right now. This is what we want to do. But is this ever going to happen is a question we want to ask. So a good example here is this is an RNA. This is from the Robert Malone's lab note, who is a famous RNA biologist. This was in 1989. He was working on mRNA synthesis and injection of rice. So this is his, and most of the time everybody thought that RNA in general had a very unstable RNA were present. So it wasn't really going to happen. We had a lot of research projects ourselves in the lab in delivery of RNA in about 15 years ago, and whenever I submitted a project on mRNA delivery through biomaterials, it was rejected, saying it's too futuristic, don't bother. We got very good papers, but said forget it. Okay, so we gave up our RNA program in 2012. We were doing gene delivery and all that stuff. We gave up all that program because it was too futuristic, had to be abandoned. This is a nice mural I like, because this implies the mRNA vaccine given in Brazil in 2021, showing that, okay, mRNA can be done. In Brazil, this is there. So, and again, the trust in science was very high. That mRNA vaccine, the superheroes were cheering the scientists, nurses, and engineers who worked on the COVID vaccine, saying that good for you that you got this mRNA to work. So the story there is, we abandoned something that was there, but 8 years later, in 1 year, we had a vaccine which used mRNA. So that's a message I want to give you, that innovation can come later on, maybe not at the time that you wanted. So future is all about the patient. It's personalized medical devices, what I truly believe. And I'm going to take that vaccine as innovation. It's a very good example to show that. So this is history of most vaccines. So typhoid fever, meningitis, this is from 1880 to 2020 scale. This is whooping cough, polio, mumps, measles, hepatitis, Ebola, COVID-2, 1 dot. 1 year to make a vaccine. And look at the other vaccines. Decades. So in 1 year, we were able to make a vaccine for SARS-CoV-2, and that too mRNA vaccine, which everybody thought mRNA project should be given up. What a miracle. Well, it's not really a miracle, what I would say. So if you look at the vaccine development process, traditional timeline, 10 to 15 years. How this 10 to 15 years was squeezed in 1 year? Obviously, there was funding, obviously, but there were a lot of parallel processes going on. The regulatory agencies, the rapid manufacturing, all the industry and the clinical trials, all of these processes were going on concurrently at 1 year, and there was a huge amount of investment going on to make that happen. And that mRNA vaccine was not rocket science, by the way. It was a simple mRNA vaccine, nothing complicated. In 1 year, this was achieved. Fantastic achievement. And if you look at what was happening at the same time, I think we need to look at that in parallel. In 2001, the first vaccine was tested in mice. Moderna starts in 2017 on an RNA vaccine trial. We didn't have COVID then. 2019, Moderna starts switching that into a SARS-CoV. So they already had a cancer vaccine going on in trial. They were struggling to raise money. Boom, this happens. 2020, we have it. So it was just reformulation, done, right? So very quick, 2020, right away, the genome was sequenced. The sequence came from China. It was faxed, literally, or scanned, done. December, US FDA authorities got the whole thing, right? So pipeline for mRNA vaccines is excellent. I mean, there have been a lot of failures also in the mRNA vaccines, but a lot of the stuff that we thought would never be done is now coming to fruition. So there's phase 1 ovarian cancer trial going on, brain cancer going on, phase 2, again, leukemia trials, all of them are mRNA, or the mRNA trial itself, a technology which was thought to be given up for zero. More importantly, this is not just a biologist or engineer's field. There's a lot of expertise that's required. Pretty much what's in prima rosa. I mean, we have biomaterials formulation, chemical engineers, regulatory affairs, infectious disease. All fields of expertise have to come together to make this happen. What I do see in future is, if I were to have a crystal glass, I would say, what are the technologies which will actually make it to market? One of them will be wearables in medtech. I mean, you have, at present we have a lot of wearables there, but I think the future is about wearables which will sense the body, the environment. First of all, the translation time for these is pretty quick. Regulations are going to be much more easier to get the wearables in there. So from the medtech space, this is definitely a personalized space which we want to see, because it dictates the environment. It's much more tangible to happen. Organ-on-chip technologies are being heavily invested by pharma companies. Not obviously academia is doing it, but pharma companies are also in part of drug discovery. They will be used for drug discovery purposes. I think it will be a much more quicker way of doing those things if they have the right models and right validation is there. They will be there, and Roche has put huge amount of money in that space. So we will see organ-on-chip at least used in the pharma industry in the context of drug discovery and drug testing. That is not going away. That is clearly going to happen. 3D bioprinting as a tool is definitely going to happen, because we do need to scale up these systems, what we call organoids in the industry. So bioprinting is here to stay, and this is mainly will be in the context of disease modeling and device testing per se. This is already in the market, meat grown in the lab. It's already in the market. But what I want to drive at, all of these technologies have come from technologies that we all have worked over the years. It's not new, but innovation will come in that space. So innovation will catch you by surprise, and COVID was a good example of that. mRNA vaccine, things that you have done in the lab will suddenly find, oh yeah, we have done that before. So so look for this space for look for innovation. So besides understanding our translational pathway and scientific breakthrough, why are these technologies going to see the light of the day? Why do we think these technologies that we work with will happen? So I talked about the scientific basis of things, and why do we think these will see the light of the day? And I really like this State of Science global report. I don't know whether you've seen this or not, but what it does is it takes 1,000 people in the population, and it takes, and from 17 different countries, and gathers that summary thereof, and then presents it. It's a really powerful way of assessing what the state of science is. And I'm going to present you some of the key facts of the latest global report. There are 5 themes that underpin the science of today. And this is again a post-COVID phenomenon. It's important to note that. One is image of science, then second is equity in science, sustainability, and shared responsibility. Hope is very important. Again, it come back to COVID. 3 years after the pandemic, the defining sentiment for science is hope. And hope, this is a nice mural I like. This is in Paris. It's in a immigrant neighborhood. And again, it talks about freedom. It's like now they've come to this space in Paris, and there is hope for them to be who they are. So I really like this mural for that reason. But all over the world, science is bringing hope for the future. So if you look at 89% of the people who were interviewed said, science gives me hope for the future. So the people are behind us, and COVID has helped it. Unfortunately or fortunately, so there is hope. And if you look at the countries that are very high, most likely is, you look at Europe as well. UK, hope is very high. Brazil, China, Australia, Mexico, hope is very high. Top 5, if you ask people what are the 5 healthcare properties, vaccines for future pandemic, obviously there's a reason why people say that. But second, we are not too far in the medical device space, cure for chronic diseases. We are right there. Cancer treatments, but is next. But number 1, this is tied, chronic diseases. Trust, where there is hope, there is trust. This is in Belfast. I like this mural because it's providing hope, because you have this superhero coming and saying, there is hope for you. I'm coming. This is the health worker in Belfast saying in the mural. And again, I have to say there were the highest number of murals done during COVID time, because there wasn't much to do, and there's really, really fascinating murals you see. Trust in science, 91%. People trust scientists. You'll be surprised what you say. People will trust you. You all are scientists here, at least in the room. They will trust you, 86%. And all of these percentages have been up post-COVID in the last 3 years. So that's an important sentiment to have. I like this mural again. This is a pensioner, and taking the vaccine because the pensioner is saying, I trust you with the vaccine. I trust what you're giving me with a vaccine that has not been tested so well. So I trust you. The trust in science is very, very high. This is in Argentina, this vaccine was done. More people will stand up to fight for science. So you have, in 2019, there were 20% who said, I'll stand up for science when debating gets made with others. But in February 21, 75% of people said, out of the 1,000 said, I will stand up for science. I will support science. I will fight against people who are skeptics of science. So again, they are more likely to stand up for science. Shared responsibility is the next one, why I think we will see this. Cross-border and public-private sector collaboration are key expectations. So this is, again, another nice mural of Einstein, which shows imagination. It's it's a whole world of imagination that we have. So science unites us more than it divides us when our views are not aligned. So science is a unifying factor for us. It unites us. There are less divisions. We can talk across people, even if we have political differences, but we can talk across them because we can talk science easily, and it's a unification factor. So that, and again, projects such as EU projects are catalysts for collaboration, for unifying that force, to understand each other more, and to make our devices that we design much more validatory or more unified in nature. And this is the most important slide, I think, is we want more science investment, and it should shape policy. This survey was not done on scientists. Obviously, as scientists, I get 100% will say, oh yes, we need more investment. This is general public saying, 91% people say, agree, investment in my science makes my country stronger. Very important. I love this slide because this is an artist in Berlin who describes media scares me more than Corona, and my take on that is we as scientists also need to manage that hype. I think our news sometimes scares me more than the science itself, because we need to manage the hype ourselves, as saying be realistic in what we say, what we can do. We are more aware on science, and science is part of the solution, and that's why I do think some of the technologies that we talk about, whether it is tissue-engineered valves or tissue-engineered bone or organ on chip or wearables, are going to come into reality because people do believe that science can make a difference. So what are my closing statements today? One is that pandemic has enabled scientific breakthroughs. We know that. We have seen that. There are a broad range of stakeholders that are involved. It's not only science. We don't have to live in the bubble. There are public. Public is equally involved in that. The patient is equally involved. We all are at stake, and we all have to do this together. Regulation breakthrough has to be important, and COVID has shown that that is a regulation breakthrough. And we need to lobby more to have this regulation breakthrough in some of the fantastic concepts that are coming out from our young researchers and ourselves. Public educational transformation, we have to use public to lobby for us. People don't want to listen to, the government doesn't want to listen to scientists, but they will listen to the public. You saw the surveys that talked about. So we have to look at public education transformation, and murals are a great example thereof. Collective ethics, it is a collective ethic of what we want to do as a future of the society. Consistent investment in science, that is understood. Sustainability is at the heart, and more importantly, personalized medicine, where the future lies. We need to move away from that concept of saying one size fits all. It doesn't. It doesn't. It has not fit all. So we need to move. We all need to recognize that even in our study designs, experiment design, we need to think about what is the gender we are looking at, what is the treatment for, what is the age we are looking for, what is the demographics, what is the race. Does this size fit all or not? It doesn't, and let's acknowledge that piece. This is a mural that we've done in Ireland, actually, which was commissioned by us. This was a co-creation of teachers, children, scientists, and muralists. So what we did was we brought scientists to the school. They talked about the science. The children drew the art, what they thought what the scientists said. We are very poor in science communication, and the muralist put that in a very disengaged part of town. So this is the final thing. But what it does embody is this is about tendon regeneration, and this shows the hand actually working with tendon regeneration, but also symbolizing hope for the device that's present. So with that, thank you very much.
We have a little bit of time for questions. Okay.
So thank you for the great presentation and also very aesthetically pleasing. I think you are living proof that even scientists and academics can produce beautiful slides. So when you talked about sugars, for example, you said that you have a lot of great tools available now, a lot of big data. We talked today about artificial intelligence. So can you say a few words about specific bottlenecks or pain points that you encounter in your field, if any, or in your line of work, for example, that if so would move your field ahead faster?
So you mentioned sugars. That's very interesting. You know, sugar. So, and without getting too technical. So if you want to look at proteins, different proteins in the body, you know, it's automated. You put something in the machine, it comes automated. These sugars, we have to manually read all of them. It's a manual process. So a process with proteins would take, say, I don't know, 2 days. For us, it takes 6 to 9 months to understand what sugars are present. So sugar is, it's not an automated process. So that's one thing. To link sugars with other entities such as metabolites or proteins or RNA is very complex in nature. So multiple experiments have to be done to understand that piece. So it's not that that can't be done, but it's not a bottleneck, but it's a time-consuming bottleneck because the tools aren't there yet to look at all of that synergy of all of that data together. It'd be great if we can get a tool which can give us the protein, RNA, metabolomic, glycomic, all those signatures together and the spatial orientation thereof. Where is it located in the tissue? So it's not only just what I get the sample, but also where is it located in the tissue will be a good readout for the design. From an engineering point of view, it'll be a design input we get. This is where the target points are. This is where we can go.
No more questions? Okay. Yes. Sorry.
Maybe thanks. Very nice. You know, that summarized what's the panel discussion finally. That we miss one important point, I think. What's the democratization of this personalized or precise medicine? It will happen easy, or we will have a little bit as in COVID, that people didn't want to distribute. You know what I mean? So that I think it's because cost at the beginning will be elevated. Then maybe in 20 years will be drop off. But at the beginning, you know, at least in certain disease application.
So I can answer the question in 2 ways, but I'll answer in a positive way. So the good thing is stratification of the population. You know, so the good thing will be that, you know, that the southern hemisphere, you know, will have its own personalization, and the northern hemisphere will have its own personalization, you know, broadly said. But that will intersect very soon, because, you know, the arbitrarization of north and southern hemisphere is changing, because you do need manufacturing facilities. Most of the COVID vaccines, by the way, were manufactured in India, the highest producer of vaccines. A lot of, you know, I mean, it was kind of a sad story that you had vaccine manufacturers there shipping vaccines outside where most of the people were dying there. You know, that is the sad story, but nevertheless, that was a transient story. You know, that vaccine spread all over India very soon. India had its own versions of vaccines going on. The same thing will happen here. If this were to happen, there will be stratification of the population, nevertheless. Just the idea of personalization gives that opportunity to personalize as well.
Thank you very much for this beautiful talk, very inspiring. The
Thank
pictures can say more than, one picture can say more than 1,000 words.
Thank you.
No? Yeah, thank you very much.
much.
And I thank, and I thank to all other speakers. I learned a lot today, from materials, reactors, to new knowledge about the body, like that we have so much sugars in the discs of our spine. I didn't know that we have so much blood vessels in our bones, and much more other knowledge. And yeah, thank you again also for all the guests who joined and listened to these talks. So we're finished with the public session. And now we have another ask from you.